at some stage you should get your feedback on the practicals, all the essential, experiments you have done yesterday. So it's me, Professor Asad, chairing this session, interim. Okay, today this is a session on drug design, structure-based drug design, and clinical development of treatments for Leishmaniasis. Two speakers, first Professor Emke, and then followed by Professor Yasu on a second talk. So I will just proceed right away and invite Professor Emke to give us the talk. So this will be more than 30 minutes or well. Not connected?
You can try to dance first. We don't want that.
Hello. Newcomers today? Newcomers? Anyone who was not here yesterday? Good. All right. Welcome. Okay, you can introduce yourselves later on, maybe. Just proceed with the session now. Thank you so much.
All right, can you all hear me? Is this working? Yeah. Well, thank you so much for actually everything. We're really, really pleased and honored to be here. I think I speak for all of us that we had a great time yesterday, and we really enjoy visiting the labs, seeing everybody, talking to the students. That's probably one of the highlights. The dance was quite nice yesterday as well, and obviously we enjoyed the food. So thank you so much for your hospitality. So I'm going to talk today. I actually changed my talk quite a bit according to what I've learned yesterday. So I've basically taken all the biology out because you know biology much better than I do. And so what I'm really trying to tell you what I would say a relatively low basis. So none of these things that I'm going to talk about, the three stories, require a huge amount of computing powers. And as you will see, programs and already said this to everybody on the table. We would like to see more questions. So please come up with questions at the end. And obviously we can have further discussions during the coffee time. So structure-based drug discovery. So the notion of structure-based drug discovery or drug design really started with the very, very first structures. The moment people saw structures on a computer screen or even on a piece of paper, it was obvious that you could use the knowledge of a three-dimensional structure to design new drugs. And this is really the first example. The second structure that was solved, hemoglobin, was used for structure-based drug design to tackle sickle cell anemia. So even the second structure solved back in the 60s, people immediately thought, okay, how can I use this structure to discover a drug tackling sickle cell disease? Needless to say, they didn't succeed. But things sort of went on, and there's now quite a few examples of drugs that are on the market that really come out of structure-based drug design. Captopril is probably the first one, an ACE inhibitor, sort of just the big breakthrough was really the HIV protease inhibitors. And in fact, I was a postdoc in Seattle at that time, and I remember the excitement on the conferences when these HIV protease inhibitors came out and really changed HIV. Protease inhibitors would have been possible without the underlying knowledge of structures and really. So great strides in structure-based drug design. Now, in principle, I just go through this very quickly, what we're going to do, and the whole concept really hasn't changed. So as a structural biologist, we need to identify a target. And as Apologies too complicated for me, so I leave that to. Once we have a target, we try to determine the three-dimensional structure. And this, of course, used to be mainly by X-ray crystallography, and Summer's going to talk a bit more about X-ray crystallography, I hope. But more recently, of course, so in best case scenario, we actually have then a structure of a protein with a ligand. This could come from known ligands. This could come from in silico methods. And I'm going to show you in one of my 3 stories how we use in silico methods to find something screening, so large scale screening. The key is that we want to have something that really binds to our target. And again, I'm going to come to that a little bit later. Right. One method that has become very popular in the last sort of 10, 20 years is finding the first ligand by fragment screening. So not screening entire molecules, but small fragments, and then taking this as an information, using the three-dimensional information to improve our leads. And again, one of my stories is about fragment screening. What is very important, and I can't stress often enough, we have to have some sort of biological characterization or biophysical characterization. So whenever we start with the target identification and try to start a structure-based structure design project, we need to have means. Oh. Hello. So we need to have means to act. Our ligand said. That should have. Ligand optimization, ligand optimization. What we find often is that sitting in front of a computer with a critical analysis and a chemist is the best way. 2 brains sort of just looking at the structure and then really trying to use a bit of intuition to, and again, talking to humans sitting in front of a computer and just replace that, right? We need to make the ligand, we need to verify our protein ligand structure, and we basically go in a circle until we come to something that is good enough for further development. So this basic idea of a circular drug development still is valid both in academia and pharmaceutical industries. So in terms of docking programs, I'm going to concentrate on a program that's developed from the Cambridge Structural Database, or probably talking to my colleagues in pharmaceutical industries and in this program is a program that the user is familiar with and knows the ins and out. So really, there isn't any program that database is that's a not-for-profit organization, and they do actually have PhD students going down to Cambridge and talking to the developers. If we have a particular problem, we can again directly talk to the developers. They do have a what they call a fair access and get free access to the program. And they told me that the University of Addis Ababa has actually a license and can use it. So anybody at the University of Addis Ababa, in principle, should be able to get a program. For the specialist, gold, big algorithm, so it uses some sophisticated for your for your program. And there's a very active development, in particular in terms of using machine learning techniques into Go. So again, it's a bit of it. Right. So everything that you're going to see in the next my 3 examples has been made and calculated with Go. So I threw this in yesterday because somebody, I can't remember, someone was talking about shell cones. So we actually have done a bit of work on shell cones on a project together with Paul. So these are small molecules that have been shown to have a very wide range of biological activities. And you can sort of take the scaffold and probably make an infinite number of molecules out of that. So Patrick, in particular, still my colleague, and Dom, they have collected redoxin peroxidase TX and PXN, right? And so they were really interested in see, can you, from the docking part, help our synthetic efforts? Now, there was no crystal structure of this, his favorite Leishmania species, Ambosensis, around there, but there was a crystal structure of a very closely related enzyme available. So we basically used a homology model. And again, today you would probably use AlphaFold to make the homology model, but the sequence identity is so high that the structure would be virtually the same. And we took all of his compounds and basically docked them into, right. I produced a number of really nice figures. So on the left-hand side, you can see the decamer. This forms a really nice ring structure made out of homodimers. And the key points, I just want to, this is up here, there you go. Cysteines. It's these 2 cysteines that are linked together, actually they're not linked together in the crystal structure. And this is sort of supposed to be the key part for the redox potential of this enzyme. So basically we can then take the homology model and run multiple compounds relatively in its on a normal desktop or laptop, and and find active binding sites. So up here is one of the compounds between the 2 cysteines, and there's another compound between the 2 cysteines. Now, Patrick was really then very keen to see, can I suggest chemical modifications based on these algorithms that might improve the binding. So the docking really binding pop. So what are the howevers? Well, one of the problems is that crystallized enzyme has both these cysteines only looked at the cysteine binding sites or cysteines. Because we assume this is where our ligand bind. But in fact, we don't know that. It could be another allosteric binding site. It could also be a cryptic binding site. So how the chemist spent the next 3 months on making new compounds, right? And then, although there's certainly a lot of evidence that this could the struct design, I would say we were able to produce really nice figures and it all makes sense, but I really would not take this as a starting point again to too many uncertain. The structure. Right. The second example is more practical, and we spend a lot more time on this because this is a protein that we have been working on together with Ari Silva up in Brazil. So cysteine biosynthesis in T. cruzi. Very briefly, just again a bit of biology. This is the pathway, and it is this enzyme that we choose as a target protein. And we also looked at the other enzyme, the acetyltransferase, serine acetyltransferase, but we never got any crystals. But the cysteine synthase was and one of the things, and I mentioned this to PhD students yesterday, is because structure determination cysteine synthases from two different species, so in order to see how different they are, and often the two different crystal structures reveal slightly different details. So the overall structures will look very similar because the sequence identity is very high and they catalyze the same reaction. But there will be subtle differences that might be very interesting. And again, I'm going to show you an example of that. So this is sort of standard molecular biology. All proteins overexpressed in E. coli, which again could easily be done in Then we are also, with Paul's help, you say to make sure that our proteins that we work with are doing. Now, I'm summarizing about 6 months of work up here. I am a crystallographer. Some will appreciate beautiful crystals up there, right? That we are quite confident that we can synchrotron. In this case, we ship them in a duo to a collection, which gives us the diffraction. When I went to my very first synchrotron trip in Stanford, one data collection took 11 days. So we had a data collection for one crystal, 11 days. The same data collection today takes 30 seconds. So all the high throughput methods that can now be applied to, or this enables all the high throughput methods. Okay. And then structure solution today, computer methods, probably a couple of days. So my PhD student was Kate, was able to solve these 3 different crystal structures from cysteine synthases, and again, as a dimer molecule. Apologies. So we always find something new. Every crystal structure. I mean, in the end, I hope, even though the structure itself looks pretty much like an alpha. So one of the things is that we found these additional binding sites, and we call these cryptic binding sites. So obviously, we know the active site quite well. We know the PRP is bound up there, but then in our crystal structures, we find other. So with these crystal structures, we then went on and sort of reliable. We had hundreds of crystal structures, and we used them fragments. So the idea is that you grow a lot of crystals, right? You soak these small molecules, they're called fragmites, little molecules, into the crystals at high concentrations, and you determine the crystal structure of every single of these crystals. So you're really going into a high throughput mode in terms of crystal structure determination. And the whole process is so automated that this can be done without much intervention, and you can basically get hundreds of crystal structures in a relatively short amount of time of weeks. Now, So again, this is just one of the examples, a bit of Now, in this case, this frag light also has a bromine atom, and Summer obviously understands why we want a bromine atom. It's just easier to locate our fragment, the fragment, and not some water molecule. So, summarizing this very quickly, this looks like this. So we basically now have our crystal structures, and we can superimpose all of the ligands, and we find these new binding sites. For example, this one in the interface. This is on active site. Then these two are possibly allosteric sites. We see there are lots of hits here and here. So this is exactly the site that we would then use as a starting point for synthetic chemistry. And in fact, we can start this also because we know exactly how our fragment is located. So again, we go to, well, our synthetic chemist, and basically ask him, how can you make this fragment into a bigger molecule? And in the ideal case, you might be able to link these two fragments. So one of the ideas is in fragment linking. So you take a fragment from here, you make a linker, and you get a much, much better ligand. So very widely used now, highly automated. Unfortunately, I can't show you the next slide because we haven't actually done the linking yet. So this is something that works well if you have actually the crystal structure. Now, multiple sites for lead development. There we go. So the last one is probably the story that is most advanced and the longest. Targets a protein from Mycobacterium tuberculosis, so it's not a neglected tropical disease, but it still kills enough people that it's worth. To give you sort of a story over the last 20 years. So this is the target, which was validated by the Boulard group at the Institut Pasteur in Lille back in 2004. So about 20 years ago, they actually solved the first crystal structure, and they validated this as a target for so-called booster drugs. And again, I'm not going to go into the biology, just take. And when we came into this project sort of 2012, they were all starting point. And we then started a new collaboration with the CCDC and really tried to go new inhibitors. So the virtual screening started old environment with a bit of Python scripts and of KNIME scripts that we needed to do, and is to identify the PDB file that is the best one for docking. And that seems like a trivial thing, but we had 12 different PDB structures. Overall, look pretty much the same. So if we go back up here, these are all these 12 structures. So overall, they look the same. So to any non-structural biologist, right, identical. But there's subtle differences in terms of how the side chains are defined and how the binding site is defined up here. So what we did is we used something that's called an in silico decoy experiment, where you basically that could bind. And then you run your strategy through this, and you want to see which of the PDB files has the highest success rate in finding your ligands that you know are ligands on top. And this is this picture up here. And to me, I think it was a big surprise that this particular PDB entry is much, much better than any others. Right? So you're selecting basically the PDB entry that has the best chance of success. And the second one we did is, so we took 6 point. Really available computational people. You filter them down from 600,000 to 6 million to 400,000. And this is done in what's called a KNIME script. Again, freely available, quite simple. And I should say my PhD student did this without any help. She just, well, got honors, read the manual and put this together. One of the things that you might see is up here, for example, this is our a volume of the binding site. So we know exactly how big our binding site is. Okay, all molecules that are larger or smaller than about 200 to 700 cubic angstrom, we throw out. So we don't waste computational time on molecules that are unlikely to bind anyway. So this brings us down to 400,000 compounds, which then can be done, well, it was done in a supercomputer in a couple of days, but in principle, it could have been done on a modern PC, right? And then the next thing is, so you basically get 400,000 compounds, and the quality of this sort of filtering was shown, in fact, that only 6 of these compounds did not find a solution. So you basically find a solution every time. And now, obviously, you can't go through all of these solutions manually. Again, there's a post-docking strategy where you basically then use the 3-dimensional information to also triage them. Because now we have binding poses. So it's not just chemical information, but we also have binding poses. About 20 criteria, and I'm just showing one of these criteria, which is called a clash score. So if they have too many clashes, right, then they're being thrown out. And the key part is, again, this is done automatically in the pipeline, and you end up with about a couple of hundred compounds that you can look up individually. So we ended up with about just under 100 compounds, which is perfect because they fit to a 96 well plate. And we used a technique called thermal shift analysis to really look at our protein ligand binding right away. So thermal shift analysis looks at the stability of our protein. And the beauty of this is it can be done on a standard RT-PCR machine in 96 well format in any lab. So anybody can do this very, very quickly. So it's a really, I would say, quick method to see if your compounds actually do bind. Out of our 96, 20 bound very well. And this a low success rate, but in terms of docking, it's actually a pretty high success rate. All right. So now I'm a crystallographer, so we also co-crystallize that. Validate our binding. We could actually validate way that the docking program actually predicted. The dynamics, I just say we did this for fun because it's nice and pretty. Doesn't really give us any new information. So this is really a success story because we could then really go and do some sort of structure activity relationship. One of my chemists longer both a better or thermal shift assay, but also a better EC50 value. So this is an EC50 value based on in vivo biological assays. So basically we can see that with these longer tails, it basically doesn't fit here into the binding site. So this is all small compound bound, and then we can sort of see there's a bit of space, but if it's too long, the binding actually goes down. It was a really nice story in terms of structure activity relationship. I'm not sure if you're yet sensing the but. It's going to come in a moment. So what was fantastic is that now my collaborator, they also worked on a different series of compounds, and they were able to get money from the Innovation Medicines Initiative with a small Swiss biotech company, BioVersus, which then also got GSK on board, and they developed a different elite compound, which is actually struggling with fun or multi-tuberculosis. So where's the but? Over the last years, there's a paper that actually came out just this year that they realized that this molecule seems to hit ETHR, our target, but it also seems to hit this molecule, which is called VRS. So they actually were able to co-crystallize it, and they could show that in the end, the target that they were working on for 15 years is probably not the most relevant target, right? And for neither of these proteins, we actually know what the natural target is or natural ligand is. They seem to have very similar binding sites. And looking at the structures, to me, it almost looks like this is a convergent evolution. These two proteins don't seem to be related evolutionary, but they seem to recognize the same ligand. From the biology, we don't know what the ligand is. And in a sense, it was very lucky and fortuitous that the ligand that the guys in Lille and GSK developed would actually hit both targets. And to me, this always tells me that biology is often too complicated and much more complicated. Sort of have to keep that in mind when different with time. Good. So so these are my stories about drug discovery and drug take-home message. I think in field. Obviously, over the last couple of years on activity predictions, and then of course de novo drug design. So I've got a few slides on that. So AlphaFold and Rosetta Fold clearly have revolutionized structural biology in the sense that we are very grateful that these validate structures. And I think that's one of the big things. Be validated by these methods. And obviously Google and AlphaFold, they put this AlphaFold database on the web with to critically analyze, and came out a couple of weeks ago. AlphaFold 3 is not publicly available. Install this on your own computer. We need to see how much of an improvement this really is. Right. But one of the things, the dynamics. So this is actually from a molecular dynamics simulation, one of our favorite. Cannot be predicted by AlphaFold, right? So AlphaFold gives us a single picture, which may or may not be true. And I mean, we've seen some structures from Paul yesterday, and it's very clear looking at those structures from Paul. You remember these transmembrane helices. I would certainly believe that the transmembrane a lot of doubt on the extracellular domains that were seen in these structures, right? And of course, these movements are important for functions of the protein. If we really want to understand protein, we also need to see how it moves. Coming back to do, right? And then predictions often see, this is my favorite structure. I went to a meeting and people told me that they're using this structure as a starting coding as protein is entirely because it's genetically disordered. Sort of a notion on where this can be used and where this cannot be used for us in structure-based drug design. There's been quite a lot of papers coming out in protein-ligand interactions where they use delta vena, which is an autodog vena, which is widely show how good they are. Right. So that's my take, and please prove me wrong. So far, there really is no clear evidence that any of these machine learning methods are better than, if we combine this with this method. Again, AlphaFold 3 can now do protein ligand. It remains to be seen. In the community, I was on a meeting a couple of weeks ago where there was a long discussion. It's a large database of in the database with different inhibitors. So for AlphaFold 3, then to find a new inhibitor should be relatively easy, right? But if there's absolutely nothing in the database, AlphaFold 3 might struggle with that. But again, it's not fully available, and I think there will be a lot of discussions and only pop. It's will be hugely biased. Just because I've got a few minutes, we did actually use which is in a program FEGROW developed by my colleague to, for our case, to find new ligands. And I looked this, I'm not going to go into detail, but I looked into this. It's called machine learning. But if you think about this, there's one little thing in machine learning in our molecular mechanics. So overall, this is a sort of a typical case where most of the pipelines are still rely on what we would call physical methods or conventional methods. And there's a little bit of snippet that's machine learning. And then, of course, it's machine learning in the title. So how much this little snippet of machine learning has helped, I am not sure. Nevertheless, I show you a few examples, and these are actually, so these are fragments grown from the cysteine synthase, so real examples. Again, it's really fast. It explores a huge chemical space. We can basically have, no, not unlimited, but we can look at billions and billions of compounds, right? But it does consider the protein as not flexible. And of course, the protein more examples up here. Oops, there you go. So there's no synthetic considerations in these programs. Again, AI can sort of come up with all sorts of molecules, but somebody has to be in the lab and make them, and also make them in an efficient way. And at the moment, that's simply not connected. So again, it's easy to take 1 billion compounds, get 100,000 compounds that bind to my binding site. How do we make these compounds? It's a completely different question. Make. Okay. Finally, really coming to end is sort of, where's AI in de novo design? My answer is it's empty. I don't know. It stays empty. I don't think it's happening. There you go. So what I think is important is really to realize that AI, particularly used sort of in the public domain, encompasses many, many different methods. And everything at the moment is called an AI, even if it's, again, just a bit of really clever statistics. So there's huge different levels in terms of what AI actually is, and one should simply keep that in mind. Okay. So as I said before, it really relies on our training data set. So no matter what the method is, there isn't a good training data set. There's no data on this particular protein. There's no data on this particular sequence. AI is unlikely to make any difference. But there's also very clearly where we simply don't know what we don't know. And those are the areas where AI might actually make play a risk. But I do think that's it's creativity of a human in the end that makes the difference. Right. With that, quick acknowledgements to my team. Funding, not going to go over all of that. So these are the people who did the work that was actually presented. And this time, as I said, I would really appreciate if you had any questions. So thank you very much. I asked every table to come up with a question.
Want to do it? We want to finish the presentations first and have a discussion later on. Or if there are burning questions, we can still press them. But,
Okay, please, one burning question.
okay, burning questions. Please make sure you ask questions. So for me, this is a learning process. This is new to me, and trying to get some insight of what you described here. So there's okay.
Okay, thank you for your nice presentation. This is very educative. One or two questions I have. Can we do this docking screening on any type of computer, or do we need special type of computer? Okay. Okay. Okay, thank you. Can we estimate some conformational change of protein structure from the software itself? Conformational change as a result of binding of a ligand, proteins undergo conformational changes. Can the software estimate that type of conformational change?
One thing we do is actually talk against multiple protein structures. So, for example, we have multiple protein structures where some side can change. In principle, we can take our ligand and dock it against each and score them. We guess it.
How many hits actually have been discovered by this approach?
Good question, but I saw a list of at least 20 very expensive compounds. So pharmaceutical industry chooses examples that are easy, like kinases, and they target cancer because that's a big market. And our challenge, we have talked about this yesterday, our challenge in the neglected topical disease field is twofold. We don't have the clearly defined targets like a kinase. I mean, a kinase is a wonderful example of structure-based drug design because there are hundreds of structures out there. We know exactly what they do, right? And we can make them in milligram amounts in the lab. So it's a sort of a really good example. So kinase inhibitors is probably the best. Aztecs is a small, well, it's not that small company anymore. They have a number of kinase inhibitors on the market. They're trying to be becoming more and more specific for a particular cancer. So one kinase inhibitor for one cancer, another kinase inhibitor for another cancer.
Thank you, MK. Just I have one question for you. How reliable do you think AI-based drug discovery is, especially in the era where we are?
How?
AI-based drug discovery, how reliable is it?
So I think it's there are areas where it's absolutely reliable, and these are so homology models, for example. So if we really take a molecule, a protein structure from something that's relatively closely related, then they're absolutely and they're astonishing good. So I would say the cysteine synthase structures where there were bacterial proteins available, the structures from AlphaFold are as good as my experimental structures. I have to admit that they're amazing. Even sort of in the fine detail, they're really amazing. And I think in terms of protein ligand interactions, they will be on the same level for those examples where you have a large data set. So for example, for kinases, because there's 100 different kinases in the database, and there's probably 10,000 kinase ligand structures in the database. So there's a huge database of known ligands. For us, the ceramide synthase that Paul represented, There's no structure of a ceramide synthase. There's only a homology model. We have no ligands. I would be very skeptical. And I think it's this kind of critical analysis that is actually bought for the young people, really not to believe it just because it came out of AlphaFold. And we see this too often in paper. So this is AlphaFold. It's the truth. No, it's not. It's a model. And we can use the model, and that's it.
Okay, thank you. I have three questions in total. The first question is how structure-based drug design minimizes off-target effects. In terms of drug discovery, one concern is off-target effect. So, in the method of structure-based design, how SBDD minimizes off-target effect and increase drug specificity. My second question is, okay.
That's actually easy. There are databases of molecules that are sort of well known to be to have off-target effects. So what you would do is you would filter your hits against those databases, and then just remove everything that looks like something that is known for off-target effects. You would filter that out. So again, you use known information to do this. Can you completely rule it out? Of course not, because they are the unknown unknowns. So it's still possible that you have off-target effects.
The second question is, as SBDD focuses on functional techniques like molecular docking and molecular dynamic simulation, these techniques hold many assumptions like rigid scoring function. And so how we just increase the reality of the drug? Because in MDs, there are many, in case of GROMAX, there are many assumptions. So how we handle these assumptions and the drug specificity and reliability?
So again, we use known information. So one thing, and I didn't show this up here, we have developed, and again, my PhD student, I didn't do that. My PhD student has developed a pipeline where you basically test the different scoring functions. All of these programs have different scoring functions, and you test the scoring functions with a known ligand. So you take a known ligand, you run it against different scoring functions. Ideally, you have more than one ligand. Let's say you have 10 ligands. And then you pick the scoring function that for this particular case works the best, because that's the problem with the scoring, and it's the same for GROMAX or other molecular dynamics simulations. You have different scoring or molecular force fields, right? In a sense, docking is a simplified force field. So again, you use known information, you run different scoring functions automatically, you pick the scoring function that works best for your particular protein. And we find this, that one scoring function works well for one protein, another scoring function is much better for another protein. And you look at the proteins and the ligands, I can't rationalize why. They look the same to me, but it's just, again, based on known information.
So we use known information, is known that.
Yeah.
The last is that SBDD is applicable for the disease whose mechanism of this is not studied and whose target is not known.
I think if the target is not known, we can't do structure-based drug design. But I think that's a problem in this field, because often people have an assumption of what the target is, right? And then it's easy to sort of take this assumption, and if you give me a small molecule and a target, I can dock it. I can show you a picture that it docks. It's not a proof. I throw that question back to my colleagues in the biology field, the geneticists, the people who do these things, and say, you need to give the target, otherwise I just can produce pretty pictures. I can still produce pretty pictures for grant applications. Very useful. Okay.
Thank you, Prof. It's very interesting. Okay, so my question is like, you have the target and then the ligand. So how do you translate that ligand to the molecule or the drug to be chosen? If you get my question, like the ligand is from some library.
Yeah.
It's not a small molecule or a drug. So how can you choose based on the ligand? How can you choose a drug? suitable for that?
So the first thing is, so all you need to give me is a chemical drawing of your molecule.
No, how do you predict the drug which can have a better binding based on the ligand?
So one thing that we can do is, so if you give me a scaffold like a shell cone,
Yeah.
right? Obviously, I can then make an in silico library
Yeah.
of all chemical modifications. And again, I would do this with the chemist together, so that we make a library of compounds that the chemist can make. Because there might be one position where you can make a lot of substituents very easily, and there might be another one where you can't. So you make this library in silico, and it could be really millions of compounds. And then he can dock all of these compounds against your binding site and score them and say, this is the most likely. And you can really, I mean, this really works. Right?
Okay.
Because if you have a good starting point, you know what the target is, you know you have a scaffold. So for example, just the scaffold of the shell cone. Right? But again, it doesn't prove that the protein is the target in the actual pathogen.
We will be breaking for tea, but we will, I hope you are happy to take more questions. Or...
I enjoy questions a lot, so yes.
Questions. Yeah, I was just informed. We should have coffee and then the presentation. Was yeah, where is she? She was organizing. Yeah, please. Meanwhile, yes.
Thank you very much. So my question is, I mean, all proteins cannot be crystallized. So what is the solution for?
So your question is, what if protein cannot be crystallized? Now we have a pretty good idea sort of the success rate. So the ballpark figures, if you use a high throughput method, like in a structured genomics project with just using robots and no brain, about 25% of proteins you can actually crystallize. If you have a dedicated PhD student who is really good and tries different things, you can probably go up to 30, maybe even 40%. But more than 50% of proteins simply don't crystallize, and we don't know why. These are proteins that are soluble and happy and everything. Honestly, I would spend no more than 6 months of a PhD student's time on that. And in particular, if it's, I would say, an easy target, like an enzyme, soluble enzyme, I would use the new AlphaFold as a starting point. The second one is that cryo-electron microscopy is really taking off in terms of size and availability and size. So membrane proteins now, as Paul alluded to, are now routinely solved by cryo-electron microscopy. And the cryo-electron microscopy is working sort of in a similar setting like the synchrotrons, that you send samples to the site. Now I know it's difficult from Ethiopia, it's actually difficult from Pakistan to do that, but I think that's something that will become more and more available. So you prepare your sample in the lab and you just send it to the national centers or to the international centers and analyze the data. So cryo-electron microscopy is going to fill some of the niche. But then there are simple proteins that will never be crystallized, will never be homogeneous enough. And I would say, again, I would go to Paul and say, give me another sample, give me another target. I mean, there's no point in burning 10 PhD students on one of those.
Maybe the second question. So we know that proteins are not static in in the body. So and these like 3D structures are a static snapshot.
Yeah.
So how do we know if it works in following the conformational change?
So one thing is that certainly there's a lot of efforts in trying to get the complexes, the relevant complexes, and particularly get them, determine them by cryo-electron microscopy. And then the times that we have sort of a single structure, a single paper, those times are over. And we actually compare different structures, already different crystal structures. So we get a pretty good sense about mobility and flexibility in different crystal structures. In fact, we never published this, but in one case, we actually compared molecular dynamics simulations and the movements for molecular dynamics simulations with different crystal structures, because we had multiple crystal structures, different crystal forms, and so on. And you can actually see that the normal modes do actually fit quite nicely. So you get some dynamic information, right? Not all of the dynamic information. And I think this is actually molecular dynamic simulations will have a big role. AlphaFold again and RosettaFold are both claiming to be very, very good at protein-protein complexes. And I've got one example where AlphaFold was brilliant and surprising. And I've got another example where we tried to look at protein-protein complexes of a human protein, where all the AlphaFold models were laughable. It was actually quite funny because they were so obviously, so completely wrong. I don't know why, we're figuring that out. But again, the problem is AlphaFold gives you a model of a protein-protein complex, and you stick it in a paper, and everybody will say, fantastic, right? You need the biology to realize, no, absolutely not. Yeah, but I think, again, for structural biology, the times that you're only a crystallographer or only a cryo-EM person or only a modeler, those times are over. We have to use all of the methods. I would say always have to do biology as well. There's a question about in vitro and biological experiments. I think you have to do that. Yeah, there's a word for the experimentalist. No, you can't do it without biology. I mean, come on, we all know that.
Thank you so much. So should we break? Yeah, because I get confusing information from the organizer. And okay, so we are to break here for coffee and 10 minutes. Not more than 50 minutes. Thank you so much.
I don't know what's
What is the camera? Okay. Okay, okay. Good morning. Unless you stop talking, I will not start. I'm sorry. Could you please stop talking? Okay, thank you then. Right. So my presentation would not be too long. Just to give you the general idea as to how generic drugs could get developed, mainly clinically. And of course, I would mention as well preclinical drug development. Oh, doesn't. It's Exactly. Okay. Okay. Just to give you the outline of my presentation, we shall look into how drugs could get discovered. What are the different ways of discovering drugs? Followed with the various approaches could be used to develop drugs. And then I will show you the paradigm shift which we have currently in drug development process. That will be followed with, just for your information, what could be the potential sources of anti-Leishmania drugs. Then we shall go to the stage of development of anti-Leishmaniatic drugs, focusing mainly on clinical development, and then the way forward. Coming to how drugs get developed, discovered, we have three ways of drug discovery. The first one is serendipitous, which happens by chance. For example, Alexander Fleming, when he was studying the various variants of cetocoxide, he surprisingly observed an area which was inhibited. That gave rise to development of penicillin. As you know, penicillin was the first antibiotic developed, and it's a very old drug. Thereafter, a number of antibiotics have been developed. So one way could be serendipitous. The second way of discovery could be spontaneous, out of frustration, out of needs. In the past years, people were suffering from malaria. They got disappointed, frustrated. Then they used to check each and every item available in the surroundings, including plants, animals, and other sources. At the end, they were successful. They came up with quinidine, which is obtained from cinchona bark, which we still use for treatment of malaria, especially quinine is the most important drug, which we use currently, especially in uncomplicated and of course complicated severe malaria as well. The third way of discovery is the one which we are recommending, in fact, which is a well-designed, a well-planned course. In order to design and to plan, you need to involve novel hypotheses. You need to know the physiology. You need to know the targets for potential drugs to bind with in order to come up with drug. And we need to think of the classic pharmacology or the reverse pharmacology, which is the paradigm shift, in fact. Then how do we approach towards development of drugs in general? First, we can make use of random screening, just like which most of us are doing currently. Okay? Simply screen from natural products certain active components which might be useful for treatment purposes, which has been claimed for. You may screen antibiotics from soil if you like. You may screen certain minerals from soil too as well. So this is purely random screening. The second way of development is through structural modification of the already existing drug. By looking at the structure of this drug, in order to come up with high beneficial effect and to minimize the adverse reactions. We can modify the structure in a way we like it and could come up with better drug. It's like amlodipine, which has been semi-synthesized from chloroquine. The third way is making use of rational drug design. In order to design it, we need to understand the biological mechanisms together with the chemistry of the drug itself. Then, currently, genetic engineering has become very popular. That is DNA recombinant techniques, in particular for protein drugs like enzymes. Insulin currently is synthesized by genetic engineering technique through biotechnology. So by changing only 1 or 2 amino acids, though it may not be insulin proper, but it does the function of insulin. And currently it's highly popular, it's available, and of course, it's also affordable. In fact, one advantage we have with genetic engineering technique is we can come up with large mass of drugs production. That's why this has become very popular these days. Then, what could be the paradigm shift? As you know, the classic pharmacology technique, if we want to develop a new drug, a minimum of 10 years, up to 15 years would be required, starting from preclinical to clinical development, which we can support, obviously. Then we need to look for the shortest way, not only in terms of time, but also in terms of cost. It's very expensive for one drug to be successful in millions USD might be required. So that makes the drug initially extremely expensive. Then we have to change the way we should go. And combining the knowledge which we have traditionally from our traditional healers, for example, and the clinical observations which you can come up with upon administering the drug which people use traditionally, together with modern technology, especially the omics technology, proteomics, metabolomics, and genomics and others, that could bring about a paradigm shift towards drug development. That is what you call reverse pharmacology technique. So reverse pharmacology technique could offer us a paradigm shift. By so doing, we can short the time to as low as 5 years to develop a new drug, starting from the preclinical assessment, going through clinical development. And it could also reduce the toxicity of the given drug, because we could have accumulated knowledge from traditional healers, which we can make use of for this purpose. And it could also shorten the time, the cost. So this integrates the documented clinical data together with experimental hits into leads. Then, how does reverse pharmacology work? Reverse pharmacology has got 3 phases to pass through. Phase 1 is experiential phase. This is a phase at which we can collect a number of documents from traditional healers or from any other people who might have used this for treatment purposes. So any observations which we have seen clinically in medicine might be starting point for reverse pharmacology. The second phase is exploratory phase, where we need to explore the dose required. Okay? How much dose do we need in order to bring about that response, that therapeutic activity? And we might need also to study how tolerable this drug could be when it's used for treatment purposes. And how about the dose range we have? And would there be any interactions if it's given together with other drugs in case of comorbidity? all this could be studied under this phase. The last phase is phase 3, which is experimental phase. In experimental phase, we can do both basic and clinical studies. But when I say basic study, we don't do exhaustively all, but we have to be very selective because we have to reduce the cost. We have to reduce the time as well. And hence, we need to be very selective. Only for those which we are not sure about, we might need to go back to animal experiments and do some studies and confirm it. And we can confirm both safety and efficacy, both from preclinical and clinical observations. Just to show you the difference between classic and reverse pharmacology. The one in the left-hand side, it is for classic pharmacology, what you call forward pharmacology. So it starts with animal studies in order to identify what effect it has, either in ex vivo or in vivo studies. We can identify potential activities which this drug could have, followed with identifying the lead compounds, the lead molecules, which will lead us to develop a new drug used for treatment purpose. And then we can identify the targets. That's why this is a very long process. It would take us a minimum of 10 years to do that. On the contrary, when we look into the reverse pharmacology, we start with identification of the target site, because we have all the information from pop medicine. So what we need to know is about the target, where this drug could interact with to bring about the desired response. That would be followed with screening the potential compounds, whether they could have those claimed activities or not. And then we can come up with back to test for functional activity. And this would take maximum 5 years. So that means then we are on advantage side if we follow the reverse pharmacology process. Having said this, then what could be the potential sources of anti-Leishmaniatic drugs? Because our main agenda is Leishmaniasis. Starting from yesterday, we have been discussing about Leishmaniasis. Then what are the potential sources where we can get drugs, where we can develop drugs for treatment of Leishmaniasis? We can have 3 different sources. The first source, screening from pathogen bugs, which our students did last, and have identified certain molecules having anti-Leishmaniac activities in vitro, and that could be possible. So this could be one potential source. The second potential source is repurposing. For example, paromomycin is an antibiotic, amino-glycan antibiotic, which used to be effective against microorganisms in the past. But this drug no longer used as antibiotic. But from the mechanism which it can bring about in bacteria, which could be extrapolated further to the parasite, then it has been found that paromomycin could be very useful for treatment of Leishmaniasis. That means paromomycin has been repurposed for use in treatment of Leishmaniasis. You can think of amphotericin B or pentamidine, and most of the drugs which we have currently, they are repurposed. The third one is the one which has been discussed yesterday by Professor Iqbal. That was from natural sources. As you all know, our country, Ethiopia, is highly rich in plants, plants, which could be used both for medicinal as well as nutritional purposes. The very good issue here is the fact that it's used for food purpose. Be very useful for medicinal purpose as well, because the dose which you use for nutrient purposes obviously is much higher compared to the one which we use for treatment purposes. So if people have been using it as food for years and years, without having any safety issue. It should not be an issue as far as safety is concerned when it comes to be used for disease treatment, because we are going to use it at much lower dose in that case. Therefore, we need to explore our natural resources we have. That's why our students are exploring to get some anti-Leishmanian lead molecule from natural resources, mainly from plant sources, in order to be used for development of anti-Leishmania drugs. So these are the three major potential sources for anti-Leishmania drugs. How about the stages of development for anti-Leishmania drugs? If we start from preclinical assessment, that is using animal model, we can test efficacy, safety, as well as kinetics, meaning how the body could handle that drug, that molecule, when it's taken. So once this is confirmed, i.e., once this chemical is confirmed to be effective against Leishmaniasis and tolerable, as well as the profile is quite reasonable, we may give it orally or parenterally or any other route, depending on the nature of the drug. Then we could proceed to the next stage. Because you might be asking, why don't we start it at the level of preclinical assessment? Why should we waste our time? Why should we spend a lot of money to develop this further clinically? Because there are 5 twos. Because there are limitations, in other words. One limitation is in animal studies, we might need large number of animals, starting from lower animals to higher animals. Because the higher the animal, the more likely to be extrapolated to human. But can we do it? Can we afford this? No, cannot. Cannot have large number of animals, in particular, higher animals like chimpanzees, monkeys. We don't have it. We cannot do this. That means we cannot confirm the result after doing in lower animals like mice and rats. As if all the results which we have obtained would be extrapolated completely to humans, which is wrong. So that's one drawback we have. The second pitfall is the time consuming and expensive nature of preclinical studies. In particular, if we do toxicity studies, the acute study might be done shortly, okay. But the chronic study, we have to look at for the whole lifespan of the animal, because we don't know what's going to happen. As you know, some of the drugs might be taken for life, like those drugs employed for treatment of diabetes or treatment of hypertension. So in those circumstances, we need to have data on chronic toxicity, but that would take a very long period of time. That is another issue we have with preclinical assessment. The third problem we have is unreliable data extrapolation from animal to human. Some of experiments not be to be done in animal. For example, HIV. If we look for developing antiretroviral drugs, obviously we cannot start with animals because, as you know, HIV is a disease of human beings. It's not animals. Okay, we cannot grow and replicate HIV in animals. So, in absence of the virus, we cannot develop any drug. We cannot see whether it's effective against this virus or not. Therefore, that is another bottleneck we have in preclinical assessment. Therefore, what is the solution? We need to come up with clinical assessments. It is a must. There's no short way other than clinical assessment to develop new drugs for development of or treatment of any disease conditions. Then that would take us to clinical trials. In short, clinical trial is any experiment. I know, wouldn't dare calling experiment because people might be very much scared. Since human beings are the one who are going to be involved in this study, if they hear the term experiment, they wouldn't participate at all. But in reality, it's experiment, obviously. The difference is experiment in humans in this case. Preclinical assessment is experiment in animals. So it's an experiment conducted in human in order to develop new intervention for future generations. If we have headache, we take aspirin or paracetamol. Aspirin or paracetamol didn't come from the sky. It's developed through preclinical and clinical development process. Then, what are the different phases of clinical trials? I know some of you might have not come across with phase 0 clinical trial. As the name itself indicates, it's not the formal trial. This is optional, it's not mandatory, but it's advisable. Because as this explore whether the one that we have seen in animal is also one not exposing certain. Not only that, not wasting food, certain for fast, not effective. Why I say it's advisable zone. The first in human is a trial. One. So phase 1 stage which we conduct in phase 0. That would be very important. Because we cannot do such thing. Because this is the first time that we can practice. So safety in animals, not necessarily safety. So we know at least heads individuals tolerate better, right? If it does happen for them. That's why we normally use. At this level, we can also study at the same time how the body, how does it work, how does it live, still live, but how it's going. So this could also be studied. Then we have phase 2. Phase 2 is purely the extension of that extension, not in individuals, but in. Because we want to confirm how safe it could be in the patient. Safe as an individual, doesn't necessarily mean safe. No. Check. Second thing is that we can prove it, whether that we can because we cannot do this in individuals, or they don't have parasites. In absence of many parasites, no absence. In this case. So this study mainly focuses on the efficacy of then once this can be vaccine at the same time safe reasonable. Then phase 3 is purely an extension of it. Use patients, but largely because under phase 2 circumstances, you may not be able to make it useful. Because the number of sample size is growing. So when you divide it into 2 or 3, then the result is obtained. And then we need to increase. But the reason why once we confirm that, we can move on. Where you get your own so it's about so this format straightforward because you can compare it with the existing drug available in the market. But the whole objective of clinical trial is to come up with better intervention. And whether the one which are superior one is a total efficacy or safety at least. So that is from all these phases, for example, free market from all these big products are going to be done for drug is market. Then once drug is market, what called is it might be wondering, why don't we know? Why don't we stop there? Once we have to forgive the drug into the market, what do we do? Go somewhere else. What's the relevance? Because in pre-marketing trial, because limited number of participants, maximum 6,000. You know, the global population lost 8 billion. Okay. Then 3,000 against 3. We don't know what's going to happen there. No? So say among 3,000 participants doesn't mean say all 8. So stop it here. Once the drug is marketed, doesn't mean only for so all over the world. We don't know what's going to happen. Same thing, nothing. It's also too narrow, meaning don't entertain any differences. Don't entertain any differences. Environmental differences. Nutritional stuff. Meaning the drug is effective in certain group of population. When this drug is taken by another group of population having different genetic makeup, may not be effective or may be unsafe. Also too short. During the trial, maybe for 6 years, 10 years. But once the drug is marketed, doesn't mean that this drug has to be used on years. You don't know what's going to happen after 10 years, because there are some adverse actions shown in data. So not able to identify, to detect the problem. Never up to the market price. Awesome. Trial size. Even in the same country, it could be different trial sites. If I put having a number of trial sites everywhere. Normally not feasible. Otherwise, other only one drug. But once it comes drug, the market is used by patient might be comorbid with other conditions. Then this patient might be protected. They don't want to buy. You don't know what's going to happen. So the two drugs need to go there. So drug will go. This case is assessment that needs in order to say what's going to happen when the investment issues. By the way, not only others, but other sharp. A very good example is minoxidil. Minoxidil is an when a post-marketing trial was conducted, patients boards started to go, hey, amazing. Might have potential to be used for clinical boards. Then, just from post-marketing surveillance, come with currently we not available. And for those who don't have a mustache, right? So that's also one of the advantages you have in post-marketing. And what we do. We have been discussing something from yesterday. Number of drugs available. That's why we call them. Oh God, most preclinical development requires here. Requires expertise, requires sources, including I think hospital safety is enough number of beds. Then shall we do that? When let's say this on behalf of the fund. Not to have this. Yeah, certain. Then what have to do there? You have to be committed. Commitment. Commitment alone here. Because these are the teamwork. We need to collaborate. We need to have our own team. We need to work from different angles. That's what we have seen earlier on about structure. You can think of a number of experts come together in order to develop drugs used for chemists, pharmacists, ecologists, physicians. You have a stake in collaborating. Commitment and collaboration are only enough. Establish a partnership. If we do this three together, then come with this
Hello, that was really interesting. Thanks so much. Of course, I have to ask a question. When it comes to clinical trials, would you do placebo against for patients, or would placebo be unethical because your drug might work and then you're not curing the patient?
Okay. Very possible. Well, placebo, not ethical. Placebo use might be ethical. If available, because not every individual has the right to privilege. So we are denying. If you put some of the individuals under placebo, as you know, placebo is for population between the parts. So we are denying them get appropriate drug. People are unethical. But like in the case of COVID, if we don't have purely drug available in the market, it clinically both will be active. Whether it's simple or simple substance, chance of it die in that case. Thank you.
So it's your turn.
So hello, everybody. Okay, you've gone through what about Leishmaniasis, the basics about that, and know how it is spreading, and then the fascinating lecture of MK, the mixed blend of structural biology and proteins dealing with that. I'm taking you towards another aspect of drug development, that is co-crystallization. I hope that all of you are aware about that. What are crystals? All you? You know what are crystals? Please say something. Okay, good. So what are the crystals? Nice crystalline material with definite shapes. Okay. So the crystalline material, shiny crystals, shiny surfaces with definite sharp edges. And whenever you saw under the microscope, they are very fascinating with respect to their colors and shapes and all other things. But with respect to a crystallographer, the crystallographer must emphasize on the internal arrangement of that crystals. Okay, so what are co-crystals? How many of you are aware about the term co-crystals? Please raise your hand. And what about you? Okay. So can you explain that what are co-crystals? That crystalline together in the same crystal lattice. Yeah, good. So co-crystals are the crystalline material in which you take the two crystalline materials or the two molecules, and they get crystallized in a same crystal lattice. As per FDA regulatory authority, the co-crystals are the crystalline materials. They are composed of two or more different molecules, typically active pharmaceutical ingredient. Usually these are the drugs or any bioactive molecule, and then co-crystals former in the same crystalline material. So during my whole presentation, I invite you to think about the API, which in my case, there is a drug amphotericin B, and then the small co-crystal former. What are the co-crystal former? The co-crystal formers are the small molecules which are recognized as GRAS material. As per FDA, the GRAS materials are those materials which are generally recognized as safe material. Means all those small molecules which are safe to be used in the human body are recognized as GRAS material. FDA has the whole list of the GRAS materials, but that is not only restricted to that material. Whenever you find any small molecule which is active and that is not cytotoxic, both in vitro and vivo, then you can consider that as a small co-former molecule. These are the different classes of co-crystals. Whenever we are talking about the crystalline material, particularly with reference to co-crystals, so they can be a hydrate, they can be a salt, any hydrated salt, solvent, or simple salt. But you need to be emphasized that whenever you have the two molecules crystalline in the same crystal lattice, there will be a no charge transfer or proton transfer within that crystal lattice. I already talked about the GRAS. So whenever you are taking any small molecule, so you must be careful about their safety and efficacy. So the story of these co-crystals started in 2007 when United States FDA approved these co-crystals as a drug intermediate. I will go through you slowly towards that why they are considered as first as intermediate and then finally approved in 2017 as a independent drug candidate. Question is that in this era, when people are exploring the various aspects of drug development, why co-crystals? What is the importance of co-crystals in the pharmaceutical sector? All of us knows very well that drug development and discovery, the pharmaceutical sector is actually, and of course the researchers as well, they are dealing with the 4 performance parameters of the drug: their solubility, their dissolution, thermal stability, and permeability across the various cellular barriers. So majority of the reported co-crystal studies are aimed at improving the dissolution behavior, means all those drugs which have a poor solubility or poor bioavailability, and thus the bioavailability of poor soluble drugs, as more than 70% of the drugs in market are with the problem of this solubility and bioavailability. So they are falling in the class 2 and 4 of the BCS classification system. This crystal engineering approach was picked up by the pharmaceutical sector, and whenever you go through the patent database, US patent database, you will find literally thousands of patents only on this co-crystallization approach adopted by various pharmaceutical companies to improve the dissolution behavior of their already marketed drugs. So what is the science behind this this co-crystallization? We use the term as crystal engineering. So what is crystal engineering is the understanding of intermolecular interaction in the context of crystal packing and the utilization of such understanding in the design of new solids with desired physical and chemical properties. So if you are aware with the crystal structure, you know that the crystallographer can provide you the insight that how the molecules are arranged in a crystal lattice and how these various functional groups are engaging with their neighboring molecules in their various interactions, that is intermolecular interactions. And this crystal engineering is that whenever you have a small active ingredient like API, let's say I'm talking about any drug, let's say paracetamol, then you must choose a co-former having that type of functional group that can coordinate with the functional group of the API. And this is the key in which they change the molecular arrangement within the crystal lattice and then contribute towards the crystal engineering of that material. So crystal engineering is mainly divided into 3 parts. The understanding of intermolecular interactions in context of crystal packing, then developing a strategy plan by which these interactions can lead to certain desired packing, and then fine tuning of crystal properties to achieve this certain goal. And this fine tuning is, you can play around by modification of functional groups of your API. So just think about that. We are talking about the drug designing since morning, even since yesterday. So whenever you're talking about any entity, chemical entity, which is actually a mixture of the two molecules. So you must have something in your mind that what would be the mode of action of that molecule. I mean, the crystal in crystalline material, there's something like that is physical mixture. So whenever you dissolve it, it will move apart and they start behaving as an individual molecule, is it? So what is the use of this co-crystallization? How they are behaving in the biosystem to improve or to deal with that your desired results? Number one. And number two, when you're talking about any bioactive co-crystal, so you must think about that what would be the use of any type of co-crystal which you designed, and that is actually not dissociate prior to reach to the active side of that active pharmaceutical ingredient. So what will be the use of this newly synthesized co-crystal for you and for the pharmaceutical sector? So these are the various questions which are still debatable in various stages of debate, and still the pharmaceutical sector and the academia and the researchers are in process of discussion on these aspects. As I mentioned, that they seems to be a physical mixture of API, and just think about that we are using a co-crystal in a topical application. So what would be the use of these co-crystals? What would be their dissolution behavior and all other things? So still, this is an area which is under investigation. And if you go through the literature, you will find every day a new finding with aspect to the mode of action of these co-crystals. So the first detail is the discussion that is related to this co-crystals and their mode of action was published as a reflection paper by European Medicines Agency in 2014. And they discuss in detail about the regulatory implementation of the co-crystals. And they clearly mention that co-crystals will not be considered as a new drug if their safety and efficacy are found to be equivalent to the active pharmaceutical ingredient of the considered co-crystal. Means if I'm talking about the paracetamol, and I'm saying that this synthesized co-crystal is more active as compared to the activity of paracetamol, then yes, on the experimental basis is proof. I say that this is a new drug candidate. But if its safety and efficacy is exactly related or equivalent to the paracetamol, then I can't claim that this is a new drug entity. It will be considered as the as same as paracetamol. Here are the few examples of various type of coformers which are already in market. Let's say this is the co-crystal of carbamazepine, that is an anti-epileptic drug, with succinic acid, valeric acid that increase the permeability across the intestinal cell. Then there is another example of quercetin co-crystals, and they have improved human cervical cancer cell activity against human cervical cancer cell activity, HeLa and colon cancer cell lines. Then in 2012, there is a journal that is Daru Journal of Pharmaceutical Sciences, and the editor wrote an editorial, and he introduced a new concept that is called as drug-drug co-crystal. And if you think about the drug-drug co-crystal is not a new concept, because your doctor is already prescribing you the various combinations of medicines to take at the same time. So he introduced the concept that why not these combination of drugs will be co-crystallized together in same crystal lattice, and then let's see what is their efficacy in the biosystem as compared to that individual ingredients. And there are several co-crystals, drug co-crystals, which are recently entered in the pharmaceutical market. Like this is a co-crystal of scubitril and valsarate for the treatment of chronic heart failure. Then Lexapro, this is a co-crystal of these two, oxalic acid and then oxalate for the treatment of depression and anxiety. And tramadol, Selexip. This is a co-crystal sold under the brand name Seglinitis, and it is a fixed dose combination of the anti-inflammatory and it is used for the management or treatment of acute pain. And there is another example. But recall that the co-crystals are the crystalline material that is composed of two or more different molecules in the same crystal lattice. We are dealing with the biosystem. It means that whenever you have taken a co-crystal or you introduce in any in vivo system, even for in vitro, when you dissolve it in the required DMS or any other combination of the solvents to test their activity. So how they retain the parameters of co-crystallization, and what would be the mechanism of action? Any idea about it? Let me give you a clue that we are talking about the intermolecular interactions. We are talking about the API and coformer. They are interacting in the crystal lattice via intermolecular interactions. So any idea about that when we dissolve these co-crystals in the solvent, so how they behave? What would be their mode of action? Any idea about it? So, the explanation is that molecules that constituting the co-crystals can retain in solution some or all of their intermolecular interactions. And because of this memory, the supramolecular complexes, although transient, could interact with target protein, enzyme, or any other entity of the biosystem by means of mechanism that is different from the individual entities. That is different from the individual API and the coformer. Means these two API and the coformer, they are binding together. There is some sort of intermolecular interactions. And because of these intermolecular interactions, when you allow them to dissolve, so they retain the memory of these intermolecular interactions. And because of this memory, they actually behave in a different way as compared to the individual entities. So, I'm presenting you some of the recent examples of our work, and the principal idea behind that present work is to co-crystallizations to study the difference in the biological properties of the existing APIs, some examples of anti-Leishmanial activity and some of the other biological activities. There is no need to talk about the neglected tropical diseases as we are talking about them since yesterday. Leishmaniasis. These are the various types of Leishmaniasis. Unfortunately, we don't have any vaccine till to date. In Pakistan, the situation of Leishmaniasis is very worst. We started with the exposure of Leishmania in 1980s when the Afghani refugees get entered in border areas of Pakistan. And since then, we are facing the problem of cutaneous Leishmaniasis. And now this disease is endemic in whole of our country. Every year from October to late March or early April, we have an outbreak of Leishmaniasis, particularly in various areas of Sindh and upper northern areas. And we are working on various strategies that we are evaluating the natural products. We are doing structural modifications by using biotransformation method, and we are doing the co-crystallization of bioactive natural products. So the amphotericin B, we all are aware that it is considered as a first line therapy in the treatment of fungal and parasitic infection. It is associated with a number of limitations like affordability, accessibility, and an array of systematic toxicities like fever, vomiting, nephrotoxicity, hemolysis, and any others. To date, it's very challenging to develop a bioavailable form of amphotericin B because of its physical chemical properties. It has very large molecular weight and its amphoteric physical nature, number one. Then it has very limited water and lipid solubility. And of course, it has a very poor absorption as well. If you see the structure of that, it is a polyene macrolide with hydrophilic polyols and then hydrophobic polyenes. And all these structural parameters actually made this molecule as a challenging molecule to deal with various structural chemists. And if you are aware about the Lipinski rule of 5, so for difference between drug-like and non-drug-like molecules. So it has molecular weight is greater than 500 delta, and they are more than 500 hydrogen bond donor, and the number of hydrogen bond acceptors are greater than 10. And all these parameters are actually making and contributing towards this amphotericin B is not available as a oral form. And these are the current formulations which are available for IV injections. Therefore, the drug availability and effectivity are the two prime barriers for successful treatment of Leishmaniasis. And what we did we co-crystallized this amphotericin B, then we evaluate its anti-Leishmanial activity against Leishmania major, Leishmania tropica. We evaluate it on A-mastigot assays as well. And we did some studies to study the effect of co-crystallization on the solubility of amphotericin B. So for doing the co-crystallization, we use a very simple strategy that we have a mixer mill. And we grind the API and the co-former on certain frequencies with certain period of time. And of course the cell is lined up with the material which is not providing or producing any heat during the co-crystallization. But keep in mind that you can do manually as well. But for manual grinding, you must be consistent, and it will take around 4 to 5 hours to take the first material to be evaluated against the formation of co-crystallization. The problem in amphotericin B is that it is not crystalline. So it means that we can't process it to confirm the formation of co-crystal by using single crystal approach. So what we did that we used the powder XRD and DSC and TGA analysis to study the formation of the new crystalline phase that confirmed that, of course, that now we have in hand a form of co-crystal. So these are the various examples that you can see that there is a co-former. And for this co-former, we study their toxicity both in vitro and vivo as well before proceeding to be used as a co-former for amphotericin B. So you can easily be seen there by powder XRD pattern that there is a strong indication of the new phase that is confirming that we have in our hand a new co-crystal in our hand. So after formation of these co-crystals, what we did that we evaluate their activities both in vitro and by using the A-mastigot assay. And you can see that in A-mastigot assay, you can easily find that the morphology of before treatment and after treatment is improved. And it is a clear indication that our synthesized co-crystal is contributing towards the improvement of infected macrophages. And thanks to our collaboration, because I had a problem in microscopy for all this, and Bartera has actually suggested me to improve these pictures as well. Yeah. So you can see easily that the morphology is improving and the infected cells are going to be decreased in after treatment. Or you can see here that this is the study of effect of solubility of co-crystallization. So you can see here that this that in some cases not only the water solubility is improved, but also the solubility is improved at 6.8 pH. That is the pH of your system. So it's clearly indicated that these are the potential targets to be explored further to study the improvement in case of both availability and biological activity as well. So this was one of the example which we use by using amphotericin B, and amphotericin B was not the crystalline material. So I picked another example, and this is which we confirm on the basis of single crystal X-ray diffraction technique. These are coumarin is a very well-known natural product. All of you are well aware, and it has a long list of biological activities as well. So you can see that in this slide, you can see that the single crystal is can easily be confirmed the formation of co-crystal, that each of our co-former is engaged with strong intermolecular interactions with your coumarin molecule. And what is actually happened that so if you can see that this is the crystal lattice, and these co-formers are actually penetrate within the layers of the crystalline material, and because of that they get engaged with their neighboring molecules via intermolecular interactions. And these intermolecular interactions are not only providing them the stability, but also providing them the various orientation of these API and the co-former, which help them to interact with the biosystem. And of course, we developed some resistant strain of Leishmania major as well. And you can see that these synthesized co-crystal showed promising activities against these crystalline material. How much time I have? No. If you want me, I can conclude it, or I have lots of examples which I can show that. Okay. Okay. Then there is an example of steroidal drug that is methyl testosterone, and this drug is a naturally occurring testosterone. It's a derivative of that, and it is synthetic in nature, and it is used for physique and performance enhancing purpose by competitive athletes and all other thing. And it also be used to treat breast cancer in women. And our main target was to explore this molecule as a candidate to active against breast cancer saline. So what we did that we evaluated for anti-Leishmanial activity as well, because we have a very large program of drug repurposing as well. And we synthesize these various co-formers, co-crystals, and you can see that with this crystal lattice that how these co-former and the API are interacting with each other. And if we talk about this in the anti-Leishmanial activity of this synthesized co-crystal, we can say that against the Leishmania tropica, the activity was not very promising, but against Leishmania major, the activity was promising. And this is the second example of this methyl testosterone, this salicylic acid. And again, the same that against Leishmania tropica, the activity was not very promising, but against Leishmania major it is. And we did some microscopy to study the effect of these synthesized co-crystals on the macrophage cells. And interesting results were obtained. And again, we did, as this is a class 2 drug, so we did some solubility studies as well, and we can conclude that some of our synthesized co-crystals have improved solubility in their structures. Here are the few examples of our microscopy, which we done on macrophage assay, and the effect of on the biological activities. This is the last example of my work, and I really love this work because this is the first example of co-crystals which I published from Pakistan, and this is a natural product. Of course, this is isolated from Seseli diffusum, and it is the plant was known for its treatment of bronchitis, asthma, liver, and we reported first time the isolation of any anti-leishmanial activity of these compounds from this plant. So what we did, that cecalin is the active ingredient of this molecule of this plant, and we evaluate its anti-leishmanial activity against Leishmania major, and then we did some co-crystallization. The debate was that this molecule was co-crystallized with thiourea, and we had a long debate with reviewers that how you say that thiourea is safe, because thiourea is generally not recognized as safe. But in our study, the combination of this cecalin with thiourea is actually contributed towards improvement of anti-leishmanial activity. Okay, and at that time, I was in Durham as well, and Paul just recommended me to do some sonication studies in order to study that what would be the effect of this sonication on the biological activities of this compound, and we did that, and we found that after 30 minutes and 45 minutes sonication, no effect on the biological activities of this co-crystallize was found. It means that the interaction is so strong that they are not moving far apart after sonication for certain period of time. So these are the results of anti-leishmanial activity, and thank you so much for your patience.
Can I ask a question? Relates to the, you took so many questions. You want to take more questions now? Is this working?
So yeah, thank you, Sama, for a fascinating lecture and introduction to co-crystallization. Just a point. The clemastine I mentioned very briefly yesterday is fumarate salt, and our metabolomic data maybe indicate that fumarate is affecting the TCA cycle as a part of the TCA cycle. So it is a bit, going back to your point of mode of action, it's quite complicated sometimes as these things dissociate. Anyway, that's just a point. So this is a question for both of you, really. So Iyasu's comparison of a pathogen box, existing therapy, and natural products. So the pathogen box is created with so-called product formulation index, or whatever it's called, PFI. So that involves C-log-P, number of aromatic rings, and a few other things. It's like a super sort of index of developability. So I would know that I think none of the existing drugs would end up in pathogen box, and probably almost no natural products because of the way GSK actually constructs these things. And also then increasingly looking at very early cutting out of things that are unlikely to be orally available, because some of you'll know this from the Donovani study currently ongoing. So I just wondered whether either of you thought that we should rescue some of these industrial compounds that have been chucked out. They're active, and maybe co-crystallization could be a way forward.
Good suggestion that we must start with some pharmaceutical available drugs to do the co-crystallization. Yeah, I'm already in process of dealing with miltefosine as well. And yesterday I got some good news from my student. Yeah, but in case of co-crystallization, the main challenge is that if you doesn't have a co-crystallized material crystal in hand, then you are facing more challenges to confirm the formation of co-crystal. Yeah, powder, yes, of course, powder XRD is one of the promising technique, but we need to do some many additional techniques to confirm that whether the co-crystal is formed or not. So I usually approaching to deal with some crystalline material. If any of the drug which is crystalline like miltefosine, then it will be a good approach to do co-crystals. Yeah.
Paul, in fact, this was my question as well to Sama. Because, as you know, most of the drugs we have for treatment of leishmaniasis are not orally effective. They are given parenterally. I was about to ask her, would co-crystallization be a solution to come up with anti-leishmaniasis drugs, which might be effective when given orally? I'm just asking. I'm not answering your question.
Yeah, I mean, if you are claiming that your synthesized co-crystal is good to be used in oral, then it means that you need to do the all procedures that is approved by FDA. I means their safety, efficacy, and all other things, because they are considered as a new drug. So you need to pass through the all barriers till final claim that this is a new drug which is used to be safe.
Tersin B itself is not crystallizable. Is there a chance for,
Yeah, that's what I'm dealing with. That we are using the powder XRD to confirm that. But whenever you are using the powder XRD as a supporting technique to confirm the formation of the new co-crystal, it means that you need to add some additional studies as well, like DSC, TG, all those techniques which confirm you that there is a new crystalline phase is introducing in your molecule.
So do you know what's the price of formulation?
I gone through that, but I mean, it's not concluded yet. I gone through that. Okay. Mm-hmm. Phase 2 clinical trial. Mm-hmm.
So I'm not sure what they did to B.
Mm-hmm.
So
I think they prepare some nano formulation of that.
yes.
Yeah, they did some nano formulation, but they did that they encapsulate this amphotericin B again by some nano material, and they are claiming that whenever it is enter your system, so it's releasing with the specific amount and something like that. So this is basically nano work. Yeah.
Very nice presentation. Thank you. My question is, for when you're using natural products as a source for drug development, one of the challenges is when you're just selecting one active molecule, you sort of lose the activity, right? So because there's some sort of synergistic activity from having multiple molecules in that natural product. So my question is really like, what's the role of co-crystallization sort of to retain that synergistic intermolecular interaction in a more controlled manner? And how many molecules can you co-crystallize? Yeah, so how many molecules? Because like I said,
Right.
Okay, thank you so much. So I have two questions. The first one is is the drug delivery system or the co-crystallization system will be faster to develop new treatments? That's my first question. Second one goes to the computational drug design. How successful is translating from the computational work to experimental? How is the success rate of translating the computational work into experimental? So how much of them, like 10%, 20%, 100% of it will be translated? That's my question. Thank you very much.
Okay, thank you for the presentation and also to give the chance. I have just two questions. One is for Prof. Samar. Is there a possibility of co-crystallization during isolation of compounds from natural product? Because we fractions our compound, then we let them dry in different environment. Is there a possibility for the formation of co-crystals in that case, that the solvents also, since we are talking about the solubility doesn't affect that interaction. So we are going to dissolve that fractions in different solvent. Maybe we use polar solvent like methanol. If that doesn't affect the nature of the solubility in polar, is there a possibility of the formation of co-crystals during fractionation and isolation of a compound? The second one is especially related to the AI techniques, drug design, actually. There is in our country, we have one issue. There are a lot of people report that this compound, a crude compound, is active toward this leishmania. Then, or there is traditionally the plant is reported as an anti-leishmanial plant. Without doing the weight bioassay, if we don't have that kind of facility, but in AI techniques, we know that if the phytochemistry of that plant is well known, and if we find those compounds in data, if we dock using the AI technique, does it working? You go my question. We have a lot of plants here. The phytochemistry is done somewhere, and the compound is known. if we select, if you study if the plant is active towards some disease, if we directly select those compound isolated from that plant and find them on AI database, and if we found that compound in the doc, does it work? Is that possible to do in this way? The other third question is actually like a request. You gave us a bioassay training there in the Korambasa, right? I really appreciate if you also arrange for us to get a training, AI training, actually drug docking techniques. Still a hand-on training. Thank you.
Okay, thank you very much, and thank you very much for the presentation. My question is about the medicinal plants. You said that in Ethiopia, you have so many medicinal plants, and you mentioned Artemisia. My question is this plant imported or it's local? And does it grow in all regions of Ethiopia or it's in some regions? And how do you conserve this very important plant, medicinal plant?
I think this question below is forwarded for me, is it? Okay. Yes, Artemisia is grown in Ethiopia, but the species might be different, okay? From what we know, the one grown in China, where Artemis name has been extracted from. So we have got a lot of Artemisia plants, and I think it can be preserved as well. I think there will not be any problem. And this is the one which we usually use for different purposes. Okay? On top of its therapeutic purpose, we even use it, as an essence, just for different purposes. For example, during holidays, people use it because it smells very, very nice. Okay? So I don't believe that there will be any problem in planting and conserving Artemisia species in Ethiopia. So Artemisia has got a number of activities. Obviously, if it's used for treatment of malaria, one could speculate this could also be used for treatment of Leishmaniasis. As you know, both have got something in common. Okay? Both are parasites, though that is Plasmodium, this one is Leishmania, which they share in common. Then there might be some common targets whereby drugs could act in order to attack any of these two parasites. So that's why, Artemisia might be potential source for treatment of Leishmaniasis as well.
About the regions where it is grown mainly, or where it's found in Ethiopia. Is it found in all regions of Ethiopia?
Origin. The origin. I'm afraid I may not be the right person to answer this. Maybe botanists should answer. I don't know. We don't have any botanist here, unfortunately. But I believe, this plant is, though the species is different, I think it might be originated.
I think Abyssinicus, Professor, yes. Artemisia abyssinicus? Afra, okay. Annoa is the main one for me.
Okay, first of all, I would like to thank all of the professors who are present this morning. I have two questions for Professor about co-crystallization. My first question is, for example, if you have three different drugs that can form maybe co-crystals, how can we predict whether the crystal is from three of the molecules? Maybe the crystals may be from one molecule. And my second question is, with regard to formulation, because crystallization is highly sensitive to contamination. So if you have a co-crystal, when you make formulation, we usually add some other additives. Don't you think additions of those additives will reverse the co-crystallization formation? So how they stable are upon storage or like? Thank you. Okay, my first question is, for example, you have three compounds. So if a crystal is formed from when you mix those three compounds, how can we predict whether the crystal is from three or from a single compound, two or three?
The last two questions for this session.
Okay, thank you. So my question is for you regarding the co-crystals. From the practical aspect, I always believe the amorphous form is more soluble than the crystal. If you can see the free energy is better than the more orderly fashion, the crystal. So how is, how is co-crystal improving the dissolution or solubility rate over the amorphous, the other form? Is there any like example in this regard? Thank you. That the case because in the with amphotericin B, I think you use I thought nicotinic. Yeah, it's I don't remember the exact structure. Yeah, does it improve because it's the structure seems more not polar.
My name is Okay, my name is Bashir Mwambi. I'm coming from Uganda. Yes, I teach at the Islamic University in Uganda.
My name is Luboa Munir. I come from Islamic University in Uganda, Kampala.
Thank you, professors. My first question goes to co-crystallization. If I got it right, if I give an example, beta-lactam antibiotic plus clavulanic acid, is that co-crystallization? A beta like penicillin plus clavulanic acid, is that an example? Okay, all right. Then in experiments during co-crystallization, have you found antagonism? I heard you talked about one experiment of amphotericin B. Have you found any antagonism when you co-crystallize? Or there is synergy, or it's just improving solubility? Then in managing Leishmaniasis in Uganda, one of the challenges is toxicities associated with amphotericin B. Does co-crystallization still prevent or contribute to reduction in toxicities? Thank you. Then my last question, AI and the development of molecules. Recently, we have had a transition because the wet lab normally has challenges, delays, and so on. And through that, through AI, in between there is bioinformatics. How do these two marry each other to complement the molecular structure to this to molecular composition to the structural composition of proteins? maybe if the sequence of amino acid is the same, rather if there are some changes in amino acid sequences, and then you have the structure similar, does that affect all you can consider, even though the amino acid sequences have some changes? Thank you.
Thank you so much. Meanwhile, I want you to comment on what they call organ on chip or intestinal, the liver, the heart. So where you want to bypass animal model, in the absence of animal models for preclinical studies. And so the industry has another model. So so they put in organs, the human organs on a chip and so on. And so I know one of our students, the PhD student, she was working on intestinal model and so on. So maybe everyone can comment on this. So do the regulations allow the use of those models, in progressing further into proof of concept studies and phase 1, phase 2 studies?
Very difficult question, Professor. I think we have to look at from ethical perspectives. Doing human intestine in vitro or ex vivo model and doing experiment, I don't believe it's ethical at all. Because we have to look at where the source is. Even for animal model, it's only in our country that we may not need any ethical approval. As you know, elsewhere, without having a license to do research on animals, you'll not be allowed, let alone human. So when it comes to human tissue or human organ, in fact, this is considered to be an ethical issue, definitely. Because, this is a sample which we have got from, or a specimen which we have got from human beings, what you call human biological materials. So for use of human biological materials, you need to get ethical approval. Unless you get it, you cannot perform. So I'm afraid it may not be possible, at least in our setting, it may not be possible to do such type of research. So the best way of doing preclinical studies would be animal model, rather. Well, mine is just for clarity. Apologies for my ignorance, Professor Samer. What are the major roles of co-crystallization? Is that to synergize the effect, or is it to improve the kinetics of that given drug? Because one of our audience asked about uh, clavulanic acid with amoxicillin. The main purpose is to synergize the action of amoxicillin, okay? Especially for those who might develop resistance. So would that be applicable for co-crystallization? Thank you.
Co-crystallization for?
Synergize.
Yeah, actually, okay. Let me bring you with the history of the development of co-crystallization. Actually, the pharmaceutical sector is the first one who opted this procedure of co-crystallization to deal with the solubility issues of the already marketed drugs. But what happened that when they did this co-crystallization, then there is a whole debate, particularly at the end of the academia sector, researcher, and of course at the end of the FDA. So what they did that they approve this co-crystals as drug intermediate. They said in 2007 they said that these are the drug intermediate which are used to modulate with the solubility aspect. But with the passage of time till 2014, it is very well clear that these co-crystals are as many of the time after their development, they are actually behaving differently with their API. So of course, you can use it for to play around with these solubility issues, even for their synergistic effect as well. Yeah. So so it's still open debate. You can find on even in various patents and then on different sites that still the people are in process of discussion about their role, about their structural aspects, and their mode of interaction within the biosystem. Yeah. So whenever you talk about the sort of their biological activities, the people are convinced that it is some sort of the synergistic effect. But still there is a whole room to prove many things which are in discussion right now.
So I think we would have to stop here. More questions than answers. I mean, this continues, and perhaps it's good. Questions here for most of us are theoretical. I guess this community will take it up further and maybe organize another high-level training on drug discovery approaches. With that, I would like to wind up this session. Please give the big for everybody. Thank you so much.
Good afternoon. So I'm going to present the result of our pathogen box screening against Leishmania atopica. A lot said about the background about Leishmaniasis. So I will stick with the treatment option. So the only treatment option that is available in Ethiopia is chemotherapy, and it's limited for mucosal diffused, complicated, and immunocompromised patients. The range of drugs available for Leishmaniasis are very limited. And those drugs have limitation in terms of toxicity, poor efficacy, inconvenient route of administration. And there is a report of mixed therapeutic responses in different regions of the country. So coming to the pathogen box, a pathogen box was a challenge provided by Medicine for Malaria Venture. It is frozen compounds, 400 compounds, provided with 5 plates in 10 micromolar solution of each compound. So the compounds have coming from different sets of disease. That means they were screened for different kinds of disease. The highest being for tuberculosis, followed by malaria, and for kinetoplastic disease sets. So this was the workflow that we followed to generate a heat map that can show the active compounds within those 5 plates. So first we prepare the stock compounds and dilute them and distribute them into 10 plates. So that means we made 10 copies of the plate. And then we have done the biological assay. The primary screening on parasites, promastigotes was done in 1 micromolar solution. And then from this, we generate the heat map. And then after generating the heat map, we select compounds which have more than 70% of inhibition. And then after gathering those compounds which have a higher efficacy, we determine their S50 on promastigotes. And then we look for the cytotoxicity on THP1 cell line and also in sheep RBCs. And then to check their suitability for intracellular assay, we have done a data mining from NSPI. And then these all informations were used to identify compounds that are safe with a better S50, that means maximum inhibition, and good physicochemical property. And then after selection these compounds, we go for intracellular assay. Then we determine the S50 of the compounds that are good in 1 micromolar solution in intracellular mastigote assay. So this is the culture media, the method, and the culture condition. So we use NNN media for test strengths and transfer to complete M1NN media. We use complete RPMI for HP1 cell line. And so this is, you can get it from different literature. So I will not dwell on this. So these are some formulas that are used to quantify the results. Cell viability was done for HP1 for cytotoxicity inhibition was calculated using this formula. Selectivity index was done using the formula provided. And to quantify the intracellular activity, we have used infection index, which means the infection rate times the parasite burden, which is calculated by infected macrophage over total number of counted macrophages times number of mastigotes per number of counted macrophages. So this was the result of the primary screening. So we did the primary screening, as I told you, in 1 micromolar. So each heat maps indicate the 5 plates, the first plate, the second, the fourth, and the up to 5th plate. And then as you can see it from the dark green light indicates that the compound has no activity or less activity, less than 50%. And the orange and the red lines indicate activities, compounds that have activity more than 70% inhibition on 1 micromolar. And then we gathered these compounds. So those compounds which have more than 70% inhibition were demonstrated in the radar graph, as you can see it from the table. So the radar graph also shows compounds with higher efficacy in red, and compounds that have 70 to 85% inhibition were demonstrated in oranges, and green color denotes compounds that have 50 to 70% inhibition, and the white radar section indicates compounds with less than 50% inhibition. So after getting those compounds which have greater than 70% of inhibition, we check for their mode of action if there is a report on a mode of action. And then we have found some reports for some compounds, and about 5 compounds, there is no any mode of action reported. So the mode of action and the reports were stated there. And then after determining these compounds, then we look for physicochemical properties from NCBI. It's a data mining. And then the purpose that the reason why we did this is because this compound should traverse to the cell membrane, also the parastophorus vacuole, to have an impact on intracellular mastigotes. So this compound should have a good absorption and penetration to the membranes. So after constructing this physicochemical property data, we compare the compounds with Lipinski's rule of 5. So the Lipinski's rule of 5 stated that compounds that have 5 hydrogen bond donor, greater than 10 hydrogen bond receptor, a molecular weight greater than 500 ADA, and computational C-log, or its lipophilicity greater than 5, are more likely have a poor absorption and permutation. So comparing this Lipinski's rule of 5 to our identified compounds, we have found one compound, which is 7812, to violate 2 of the Lipinski's rules. Rule 1, which is molecular weight greater than 500, and rule 3, which says a hydrogen donor, a hydrogen receiver. So it violates both the 2. So it's greater than 10 and greater than 500. So it was disregarded from further investigation. And then we go for intracellular assay. We screen these compounds at 1 micromolar first as a primary intracellular assay. And then we just found only 6 compounds active at 1 micromolar. And then after that, we go for IC50 determination in intracellular mastigote. So the most potent compound in intracellular mastigote assay was 90102, which is also very effective in previous anti-Leishmaniasis activity. This compound is reported as a hydrofolate reductase, and so this is the most potent one. and followed by, I think it is 5071. So this is concentration, or the IC50 is stated as a nanomolar concentration. So it's not in micromolar. So all of them are in nanomolar. So the IC50 of 102 is was comparable to the IC50 of amphotericin B, because we have found the IC50 of amphotericin B around 78, something like that. So it indicates that this compound might be a good lead for Leishmaniasis epidermolysis. So coming to the last slide, the recommendation. So we make the, even if it's a low throughput screening, we made it in promastigotes. So this method might miss a compound that might have activity on intracellular amastigote. So we recommend that this compound should be also screened in intracellular amastigote in high throughput assay, because we might get compounds that might missed on the promastigote assay. So the pharmacokinetics and safety evaluation is needed for those compounds which lacks this kind of data, biological data. Mechanistic studies should also be conducted for those compounds, because most of the compounds, they don't have a known mechanism of action, and their toxicity and efficacy in pharmacokinetics in mammalian cell activity should be investigated. So two compounds on primary assay were very effective. They were like in like 10 nanomolar concentration and something like that. So they were very effective in promastigote assay, but they lack their effectivity in anti-amastigote assay. So maybe we should look at these compounds and look for a structural or remodeling or something like that. So in vivo animal models should be considered to determine necessity of bioactivation of these compounds and provide insight into their safety and efficacy that can give us the full picture in a living organism. Thank you.
That's one question. Discussion. So this was prepared in our lab yesterday. It's a lot of work. No, it's the stage.
So you sort of briefly touched on it, but you suggested that maybe starting with promastigotes might not be the best way. We've noticed some compounds having actually no activity against promastigotes, but having activity against amastigotes. I thought the pipeline looked very strong and well designed, so well done on that, very clearly described. But do you think, in retrospect, you would start with azenic amastigotes and then work into infected cells?
Yeah, that's my point. So if like the materials and all things are available, so it's better to start from the amastigote, because when we see the probability of getting a good candidate by screening for promastigote or amastigote, there is a high probability of getting good compounds if we screen them from the amastigote stage, whether it's axenic or intracellular amastigote. So we should consider that one, because it's like 400 compounds. So a promastigote assay is like a bit easy for us to screen them. Yeah. Yeah.
But it's a mastigote stage starting there is a challenge. It takes 7 days not to check infectivity, cell, you know. So that's true. For instance, pentavalent antibody, you can do the assay in one stage only, but when you do the trivalent, it's fine for the stage. So it can happen. That's okay. Move on to the next presentation now. Oh, Bashir, sorry.
Islamic University in Uganda, yes. In Africa, there are two Islamic universities, and there is Anglo and and one in Uganda. I think one is in Cameroon and the other one is in Uganda, French and the English. So the English is in Uganda. This is the Anglo Islamic University in Africa. So this one is in Uganda. My name is Bashir Mwambi, and I'm going to go through a review of a neglected tropical disease in Leishmaniasis in Uganda. Next. Okay, the methodology is a review. We have looked at the different works that have been done from several scholars and Ministry of Health to come up with this challenge. And to start with, two common forms of Leishmaniasis, but the commonest in Uganda is Leishmania, rather is the visceral Leishmaniasis, which inhabits the northeastern part of the country. And historically, this gives us a basis why it is a bit neglected. First identified in 1951, and after that, in Karamoja region, 4 other cases in Karasuk were reported. By then, it was Kenya that was managing Leishmaniasis, so that those cases were taken to Kenya, and then a further 2 were identified. That was the start of treatment of Leishmaniasis in Moroto. Then later, more cases were reported in the Karamoja region, and the challenge then was they were confused by other liver diseases, which included hepatitis and malaria. At that point, they were managed as complicated malaria, and many people succumbed to Leishmaniasis. Most of them were identified as rather after postmortem. Now, at that point, the work on Leishmaniasis stopped for about 4 decades. It was later on in the early 2000s that again, the signs and symptoms came to be familiar with scholars that have reported that the work of Leishmaniasis was reignited to be supported in the eastern part of Uganda. Now, it is at this point that Uganda reports the cases of Leishmaniasis, but earlier on, there were cases being reported in this region. The current epidemiology as of 2000 to current, the common cause of Leishmania is Leishmania donovani. Most of the work that has been conducted has reported Leishmania donovani. In some of the work that we did with the Medicine Sans Frontier, characterization gave Leishmania donovani, and entomological studies showed the Phlebotomus martini as the cause of Leishmaniasis, but there have been reports of vertical transmission of the disease. And the Karamoja region, as you can see on the left, that is the north part of Uganda, and the comprise of quite a number of districts, Moroto, Kotido, and others. So near Kenya is where the Pokot group is, and this is where commonly the disease is. And where the name Uganda is the metropolitan Uganda. And there are reasons why I'm projecting this is most of the decisions are made around the central, and the other part is quite remote. It's because it is a semi-arid area. Many health workers don't want to go over there, so it is quite neglected. The area itself is neglected, and the contents. And when it comes to health sector strategic plan, this area is not always taken care of. And we find that a number of people normally succumb to Leishmaniasis without management. The other, the tribes that inhabit this area are the Pokot, the Pian, Matheniko, Bokora, Tepeth, Jabol, and Jie. And there are studies that have tried to characterize whether Leishmaniasis is strictly, or there is any genetic predisposition of Leishmaniasis to this group of individuals compared to other part of Uganda, though there has not been any difference in genetic susceptibility. That leaves us with the environmental factors responsible for the endemicity of Leishmaniasis in that area. It should also be noted that it is in this region that we have trypanosomiasis. There are two forms of trypanosomiasis caused by Trypanosoma brucei and the Trypanosoma brucei rhodensiensis, which inhabit this group. Some say it's because it is a cattle corridor, but others say it's because it's still a neglected area. Now, visceral Leishmaniasis at a glance in a country. Locally, the people call it Tamiz. I also don't know what Tamiz means, but it is a local name. I can't translate it, but it has been translated into enlarged spleen. And typically the enlarged spleen in a country from the central region, which is infested with malaria, has always been taken as severe complicated malaria. And that is what has been managed. Later on, the occurrence of post-Kalaza Leishmaniasis opened eyes to the health workers and the start of management of Leishmaniasis. And the diagnosis the diagnosis is a bit challenging because of lack of specialized health workers in the region. Traditional microscopic examination of tissue blood samples from bone marrow, the spleen, aspirates, lymph nodes, being the gold standard, but sensitivity is quite low. This would be improved if at all there is an extension of the molecular diagnostic assays in the region, but the disease itself as Leishmaniasis hasn't been taken into account. It is at treatment that the Drug for Neglected Disease Initiative started. Okay, this has tried to do some work, but this started around 2016 to try to give management and implementation of research in Leishmaniasis in the region. Other areas that are supported by this, that support Leishmaniasis include the MSF, Swiss, and this basically was to try to teach how to manage Leishmaniasis in the area until when the country in 2016 adopted a treatment regimen. The treatment regimen first is a combination of sodium stable gluconate, IM or IV, together with the poromycin. But there are individuals who are who have contraindications to poromycin. That means the period is the duration is improved from 17 days to 30 days using sodium stable gluconate, and that is also a challenge because it's only injectable. There are no orals. People don't want orals. Then in pregnancy, this area was also affected by hepatitis A, rather hepatitis E, and the coincidence of hepatitis E and Leishmaniasis contributed so much to mortality, and there was optimization of treatment for use of liposomal amphotericin B, still IM, and in HIV, it's liposomal amphotericin B that is given. The challenge comes when it is post-Kalaza Leishmaniasis, where treatment has to be prolonged and the treatment success is not always good. Why? And trying to assess a single drug to a combination that has been done. Okay. So, and in doing so, there was an assessment recently of a combination of drug versus a single drug, and it was found that there was no difference when managed by a single when the disease is managed by a single drug, except for age, that those aged less than 15 years and 45 and above had the parasitemia clearance quite lower compared to the mid-age. Thank you so much.
Question. This is on the name. The vernacular term is associated with the termites.
Mwambi.
There is this species called Macrotomus termis.
Oh.
So that, people generally stay around the termite. They stay in the termite.
Yes.
I know all of this. So there must be another term for the spleen itself, larger spleen. Everywhere we have a name for it.
Yeah, it means the ball.
Yeah, something. They must have.
Yes.
63.
Yes.
Terms. So any question for Bashir? If not, okay.
So thank you for that overview in Uganda. Just have you noticed in your work and your review the number of cases rising, or is it unclear how many cases there actually are?
There are no epidemiological studies that take on. It is rather doing researches in Moroto Hospital. It's particularly one hospital that research is being done. Whenever we want to do research, you go to Moroto, do some work, but not covering the entire region. If you look at the region, it's only one district that is assessed, but the rest are not assessed.
Thank you. So I'm Dr. Abdurrahman Surur. The topic of my presentation would be regarding fexinidazole. Yeah, so for the times this project was funded by Alexander von Humboldt Foundation, and yeah, so for the time of sake, I would start with what fexinidazole is. As you might know, fexinidazole is a recently approved drug for human African trypanosomiasis. But this drug also this drug also was found to show anti-leishmanial property both in vitro and in vivo setting, which is not surprising considering both of them belong to the kinetoplastid class. So because of this, the clinical trial of fexinidazole for leishmaniasis was started. Fortunately, this was interrupted, and the reason was with the exception of 3 patients out of the 14, most of them, they show relapse. So because of the activity-related issue, the clinical development of fexinidazole was terminated. So the first problem of repurposing fexinidazole for leishmaniasis is activity. The other ones are liver injuries, in that the ALT and the AST concentration was 3 times higher than the upper limit normal in 13 to 15% of the patients. So liver injury could be the problem that we need to tackle. And the other one is the heart rhythm problem, in that the metabolite of fexinidazole was found to inhibit a channel called heart channel, which may lead to the change in the rhythm of the heart. So we need to tackle those 3 properties if we want to forward fexinidazole for leishmaniasis. So how what fexinidazole for leishmaniasis is, we have to know the structural features which are important for the activity and those which are responsible for the toxicity associated with fexinidazole. There is very little information known in terms of the importance of the structural features for activity. For example, only 2 metabolites have been identified so far, which are the sulfoxide and the sulfone metabolites. But if the predicted metabolism holds true, then there could be other, like for example, the nitroso intermediates, there could be the hydroxylamine, there could be the amine, all of them can be responsible for the liver injury associated with fexinidazole. So we need to uncover more into the structure of fexinidazole so that we can establish which structural moieties are responsible for the activity, so that we can establish the structural activity relationship. And we can also find the moieties which are responsible for the toxicity, so we can develop these structural toxicity relationships. After that, we can do the modification we need in order to enhance the anti-leishmanial efficacy, as well as we can remove, for example, the nitro group could be responsible for the hepatotoxicity, so we have to find other moieties if we want to remove the hepatotoxicity profile of this fexinidazole. We have to do the same for the HERG. So initially, we did the synthesis with a little modification to the published procedure, in that we started with the less cheaper starting materials, and we tried to reproduce both of the metabolites in one scheme. After that, we evaluated the anti-promastigote activity of fexinidazoles in the strain which are already reported. In addition, we include some strains of our interest. For example, we included Leishmania donovani, which is resistant to miltefosine. So if you can see the data, fexinidazole, which is number 1, it has very good activity in all strains of the tested, like Leishmania mexicana, amazonosis, and donovani. But its metabolites, they are at least equipotent with the parent compound. And surprisingly, both the parent compound and the metabolites, their activity was much better than miltefosine, which is the latest anti-leishmanial drug available. So this gives us like a good starting point to use fexinidazole as a prototype for further chemical optimization. So we synthesized various analogs. For example, we synthesized the sulfoxamine analog, because sulfoxamines can improve the oral bioavailability more than the sulfoxide and the sulfone. And they can also give us more hydrogen bond interaction because they do have mono as a group. Unfortunately, the activity was not much better with this analog. The next modification we did was to replace the sulfur with carbon, because as you can see, the metabolites are the sulfoxide and the sulfone. So if we change the sulfur to carbon, then we can block the metabolism, which is very important because fexinidazole is completely converted into its metabolites within 1 hour. So we try to synthesize the carbon. This gives us like a very similar activity to fexinidazole, but the problem was its hepatotoxicity profile was a little bit higher than fexinidazole. So we didn't want to pursue this analog. We wanted to know if the nitro group is essential for the activity. There are some data which I saw, but as I showed earlier, its metabolism is a problem. That's the reason why nitroaromatic drugs are rarely featuring into the contemporary drug discovery. So we replace it with hydrogen, and analog 6 was completely inactive. But this could be due to the hydrogen cannot maintain the size or the electronic feature of the nitro group. So we did isosteric replacement with the cyano analog, but still the activity was lost. So this could be that the nitro group is essential for the activity. We tried to replace the linker. For example, we tried to replace it with amine. This leads to a reduction in activity. The other analog we synthesized are the stable carbon analog, because whenever I try to reduce fexinidazole, what happened is the ether block always cleaves, which is not good, because if the ether is blocked, then the metabolite is phenol, which can be oxidized and could be responsible for liver toxicity, just like observed with paracetamol. So we wanted to block that, and I synthesized the stable carbon analog, and I improved the solubility with the sulfoxamine analog because the 9 was a little bit hydrophobic. And we synthesized also some hybrid, in that we wanted to couple the structure of metronidazole with fexinidazole, because metronidazole is also reported to show some anti-leishmanial property, and they have similar 5-nitro imidazole scaffold. So we synthesized this analog also. So this is the scheme. I would not discuss. So if you have, we can see it later. But all of this analog, we couldn't find an analog which has more improved activity than fexenindazole. So we went to next set of modification, and we focus on drugs which are used currently for trypanosomiasis, like nifortimox and others. So they do have amide bridge. So we replace the ether bridge with amide, which is also more stable. So this is another important, but yeah, we synthesized two analogs. Also, we wanted to see if the methyl group is important. So we synthesized the analogs without the methyl group as in analog 13, but both of them were not active. The other modification we did, we used the hybrid strategy in that we replaced the aromatic or the planar phenyl group with the thiomorpholino group. This analog was relatively active. The other modification we did was there is a recently approved investigational drug called acoziborole for human African trypanosomiasis. So we split the structure of acoziborole and we coupled it with fexenindazole. So we synthesized 3 analogs this way. It's a bit hydrophobic, so we did self-oxidation to improve that one. And this one also contained the other side of the structure with 2 trifluoromethyl 4-fluoro analog. Those analogs were relatively better in terms of activity, yeah, but they have other problems that I would mention later. So the next strategy, what we wanted to use was to use bicyclic strategy. It's a very old technique, but still nowadays it's a trending topic, and drugs like delamanide and proteomanide has been introduced with this strategy. So we coupled or we conjugated the fexenindazole with pyrazinone structure, and we synthesized 2 analogs. So this was the reaction scheme we used. Unfortunately, these compounds were not interesting in terms of anti-leishmanial profile. However, we synthesized other conjugates that the conjugation of nitroimidazole was oxazole was reported in delamanide and proteomanide. So we did that with fexenindazole, and we synthesized the racemates, the R and the S enantiomer. We synthesized the sulfoxide, the sulfone. These were very interesting because their activity was much better, as I will show them later. So the other, it's not only activity, we wanted to see the metabolic stability. So we use microdomal incubation, S9 incubation, because as I mentioned earlier, fexenindazole undergoes very rapid degradation. So this way, some of our analogs, for example, analog 20, 21, 22, which shows very good activity. As you can see, after 32 minutes, there is significant amount left. But if you see compound number 1, which is fexenindazole, nothing would be left after 32 minutes of incubation. So we found analogs that do have better stability than fexenindazole. We also evaluate their cytotoxicity. Most of our analogs were not cytotoxic, with the exception of some analogs, like analog 5, 13, 15, and 18. We also evaluated their hepatotoxicity using MTT cell viability assay in TAMHA cell line and HepG2 cell line. With the exception of borolactones, most of the drugs, they did not show any acute hepatotoxicity profile. We also evaluated the hERG safety of our analogs. Here it was a bit interesting because in literature it's reported fexenindazole does not reduce the hERG activity, but our assay shows it's even it could be potent with IC50 value around 12 macromolar. It's capable of inhibiting hERG activity. So which was a bit interesting because in the literature, only the sulfone metabolite was reported to do that. But in our case, we found fexenindazole can also block or can also reduce the hERG activity also. So the activity, as I mentioned earlier, the activity of compounds 10, 20, 21, and 22. So as you can see, the first four are the anti-promastigote activity. And if you see the first one, fexinindazole, it's active. But if you see 20, 21, and 22, 20 is a racemate, 21 is the enantiomer, 22 is the enantiomer. They do have several fold higher activity, anti-promastigote activity compared to fexinindazole. And if you see the anti-amastigote activity, which was done in LD miltefosine resistant Leishmania donovani cell line, they do have very good activity, like 0.4, 0.6, and 0.4, which is compared to the parent compound fexinindazole, does not have cellular anti-amastigote activity. It has axenic amastigote activity, but not intracellular amastigote activity. But this compound is showed very good anti-amastigote activity in both wild type and resistance Leishmanial strain. If you can see the microscopic image, so this was with miltefosine. If you increase up to 20 macromolar, still it's infected with the amastigotes. But with our compound S29 with 1.5 macromolar, almost very few amastigotes was left. So this shows our compounds have very good activity. So from all compound we synthesized, analog 22 shows very good activity with IC50 value around 0.4. It has better metabolic stability. We were not able to see any reduction in hERG activity up to the maximum concentration we were able to test. So it seems analog 22 has all the favorable activity that we were looking for. So thank you.
Thank you very much. Very nice talk. I was just wondering, what do you know about the mechanism of fexinidazole? What is known about the mechanism?
So there is only a proposed mechanism. The proposed mechanism is this is the proposed mechanism in that there is an enzyme called nitroreductase 2, which is responsible for the activation. So it's, like I said, it's proposed, it's not exactly known, but it's believed that the nitro group will be reduced by this nitroreductase enzyme, and it will lead to the generation of reactive species like the nitroso metabolites or the hydroxylamine or the amine. All of them are capable of making a damage in order to inhibit the DNA synthesis.
All right, so the next presenter, Leishmaniasis control.
Okay, thank you very much, Protocol Dubbed, and good afternoon. I'm Lubo Amnir. we are to look at leishmaniasis control in Uganda. In other words, the achievements, what has been achieved, and the challenges they're in. IU, as stated before, it's Islamic University in Uganda. As we know, that leishmaniasis was first detected in Uganda in early 1950s, but it was not much considered, and it has been much considered in 2000s. So the prevention and control that have been much taken by Uganda, the government of Uganda, I find that one of them is insecticide treatment of bed nets to control sandfly bites. And this is in conjunction with the malaria control program. You find that the government has been able to distribute malaria, in other words, mosquito nets in so many districts in Uganda. And you find that in those areas that are endemic with leishmaniasis, you find that this malaria control program distributes nets that are treated with both with insecticide that can kill both mosquitoes and the sandflies. So additionally, you find that indoor residue spraying with insecticides has been implemented. You find that all the indoor residues in the endemic areas, they are sprayed with insecticides. So public health education campaigns are carried out on radios, televisions, as well as community radios in remote areas, so that they can sensitize individuals and they can control, and thereby leishmaniasis can be controlled. So doing all that, you find that there are so many challenges that this program faces. And you find that the first challenge is inaccurate initial diagnosis, whereby other symptoms are always vague, leading to misdiagnosis in some cases. So you find that some cases are mistakenly considered to be brucella, and they are managed as brucella, yet it is just leishmaniasis. Molecular methods are not used much as the country has embraced molecular diagnosis. So the country has embraced molecular diagnosis, but you find that it is not much used in so many areas, and most especially in this endemic area, because it is a remote area and much research has not been done in this area. So you find that even the molecular diagnosis is not much used, yet the country has embraced it. Where competent health workers lack training. You find that the health workers lack training. If they are trained to use these molecular methods, for example, they can easily manage this leishmaniasis. Limited accessibility to diagnostic tools. Some tests like spleen, bone marrow aspirates, you find that requires skilled personnel and are not available in remote areas. We find that using these tools, it requires skilled individuals, and these individuals are not available in those remote areas. As it has been stated before, that the hospital, the only common hospital that manages this leishmaniasis is in a remote area somewhere, but other remote areas are not much considered. You find that there is even limited accessibility to those to that hospital. If you get it from another district, you cannot easily access that hospital for diagnosis. So there is also inability to detect asymptomatic cases. Existing tests may not pick up asymptomatic infection or predict progression to full-blown Leishmaniasis. So we also have a challenge in treatment whereby limited drug options are available. So you find that the current arsenal of drug against leishmaniasis is relatively small, raising concerns about overuse and a potential drug resistance. In this case, you find that this raises potential drug resistance, drug access and the cost. You find that the effective medications are expensive. If the NMS, National Medical Stores, do not procure these drugs, you find that it is very difficult to access the drugs unless the National Medical Stores intervene. So you find that treatment side effects is also another challenge, whereby current drugs cause unpleasant side effects, reducing patient compliance. For example, amphotericin B, which is associated with so many toxicities. We also have a challenge of treatment complexity. So treatment regimens require lengthy hospital stay and involve multiple injections, which can be difficult for patients. You find that patients cannot, in most cases, they do not adhere to this. So post-causal dermal leishmaniasis treatment remains unsatisfactory. So currently, you find that the immune response is implicated, but the parasite persists due to drug resistance and possibly also due to nutritional deficiencies, which is the biggest challenge of the region. You find this region has a problem of, or a challenge of, nutritional deficiencies. So what about the prevention and control? The challenge of prevention and control. You find that people in this region have limited knowledge about the vectors, since it's a remote area, and yet the central district, the policy-making districts are within the central, and this is in the far northeast. You find that people in that area find it difficult to access knowledge about this vector, the sandfly. So socioeconomic factors whereby generally marginalized, yet literacy rates are the lowest in the country. You find that when you consider the whole country, the literacy rate in this endemic region is very low. So you find that this area is marginalized. So what about the poverty? You find that people in this region lack proper housing, proper hygiene, which create breeding grounds for the sandflies. Also, there is a challenge of climate, whereby this area experiences harsh climatic conditions with torrential rains and floods, and these are later accompanied by high temperatures and a drought. And this favors the breeding of these sandflies. So, locality of infection, the disease is in one region, and that region is remote from the center. In other words, the decision-making districts, it is far away from the decision-making district, So because this endemic region is located far away from the decision or the policy-making districts, you find that it is neglected, and the location of that district puts it at high risk, puts the individuals at high risk of getting leishmaniasis. Bayer is centered in the health sector strategy plan. So in conclusion, you find that visceral leishmaniasis is apparently more widespread in the northeastern part of Uganda than originally thought. We find that it is much more as compared to what was considered before. So first-line drugs have now been registered in the country with distribution challenges. There is also a problem in distributing those drugs. Management of visceral leishmaniasis is well established in Amdat Hospital, the hospital that I have mentioned so many times. You find that it is the only hospital that is managing this leishmaniasis. You find that through the prescription, sustainability and wider coverage remains a challenge. You find that though the prescription, sustainability and wider challenge remains, and the wider coverage remains a challenge. Research and surveillance on leishmaniasis is still weak. There is not much research on leishmaniasis except in just a few, except by just a few researchers. Visceral leishmaniasis is one of the neglected tropical diseases in Uganda. So recommendations, as we recommend, we find that strengthening the capacity of local institutions to conduct surveillance and research, also to promote effective management of visceral leishmaniasis, especially post-causal dermal leishmaniasis. Then collaboration for improved surveillance, diagnosis, treatment, and prevention is a key. So collaboration between health institutions and research institutions is also required, and increased funding for control programs is also very crucial. Thank you very much. Shukriya.
Where is the geographic location? Yeah. Where? Okay. So it's like sort of increasing overview of the challenges that we are. So if you could choose one thing, of all the things you mentioned that would have a rapid impact, what one thing would you choose?
Actually, we need much research, and then we we approach it at a molecular level. We need to approach leishmaniasis management at a molecular level. That's what it can give a high impact.
Thank you so much. Then we have the last speaker now. So, you're ready? Like I guess. Yes. Oh, that's okay on this one, yeah. Right. Chief.
Okay, thank you. I'm Grandma. My talk is on validation of in-house liquid direct activation tests, antigen for the purpose of diagnosing viral Leishmaniasis in hotspots in North.
How's the
Serbia.
speaker? How's the speaker?
Okay, this is my presentation outline. It comprises introduction, methodology, result, and finally conclusion will be made. So I will skip this parts, introduction parts, and directly go to the diagnostic parts. So viral Leishmaniasis is usually diagnosed by different mechanisms. One is parasitical diagnosis, which includes microscopy and the culture. And the second is molecular, it's just PCR based in the lab. But when we see these methodologies is usually involves invasive sampling techniques, and it also needs specialized training, and even it needs expensive setups to conduct these experiments. Thus, even this is less common in most resource-limited countries, just including Ethiopia. So the other detection mechanism is serological tests. The first one is direct activation test. The second is RK39 based, and third one is indirect immunofluorescence antibody, and the enzyme linked immunoassay sorbent is the serological or the indirect mechanism of techniques. So that and RK39 is usually used in our country, and this is technically less demanding, and even it involves minimal or non-invasive sampling techniques and better sensitivity and specificities. So even in our national guidelines, this RVT, RK39 and that is recommended for the diagnosis of VL. Among these even serological tests, that was shown to be more sensitive and specific for the diagnosis of VL in endemic areas. So the rationale of this study was national programs is highly depend on donors just to purchase this data. So this which will lead to unnecessary delays due to import regulation or processes. So based on these backgrounds, the aim of this study was to prepare in-house data antigens and to compare its diagnostic performance against validated techniques. So for this, for the validation techniques, we used different samples. So our sample source were from confirmed VL cases, around 110, and non-VL cases, 86, and other, we used 76 apparently healthy control as a controls for the validation of this test. So at least these materials are required for conducting or for the preparation of these data antigens in our lab. So this is how we did the antigen preparation in our lab. So the first step is just culturing of these parasites in lit media, which is it is liquid media. Then we tried to, after we culture that it needs mass culturing, so high number of parasites is needed. So this mass is collected in a falcon tube or blue tube, and then centrifuged at 300 revolution per minute. So for 10 seconds. And after this, we just washed 3 times in log solution. And then we sediment the parasite and discard the supernatant, and the sediment then diluted in log solution again, and then washed 3 times. So after that, we add the, actually it's just, I'm in difficulty to reading, it's very small, is in small, is written in small. So the 5th step is just incubation with 2 mercaptop ethanol alcohol. After that, we centrifuge and sediment the parasite, and this sediment was just fixed with formaldehyde, and then the fixed sediment is the supernatant is fixed in formaldehyde, and then the sediment is suspended in a 50 ml log solution, and then in 100 ml of capacity of bottles. So this solution is just mixed on magnetic stirrer for 3 hours, and then the sediment is again incubated at 4 degrees centigrade overnight. So in the next day, we centrifuge this solution, and the supernatant is discarded, and we add the sodium citrate solution. So this sodium, the sediment is then after just centrifuge this in sodium citrate, we add in just we stain these solutions using Comase blue or crystal blue solutions, and this after we just rinse 3 times with sodium citrate, we add the fixative formalin. So this formalin is just fix the parasites. So after this, we used these parasites for as antigen for the next experiments. Okay, this is how we did the dot testing. So in the first column, we add our diluent antigen, our diluent, and the reagent or the dot antigen. So the second row, we add our controls and samples. So first we add the diluent. This is gelatin saline. So we add 100 ml, 100 microliter of our diluent in the second row. But in the first row, we add 50 microliter of gelatin saline with mercaptoethanols. So after that, we add in the second row 100 microliter of our diluent, and next we add 50 microliter of gelatin plus that. So the first row is contains or comprises our antigen and only the diluent. So it's used as a control for the consecutive reactions. So the first row is we add our negative control, and in B2 we add our positive controls, and on C2H we add our samples. So after we add the sample, we serially dilute our in this 96 micro tetraflate. So in the second row, we do have 100 ml of 100 microliter of the solution. So we dilute serially, and finally 50 microliter will be discarded at the end. So when we look our dilution, there is a cut-off point. So 1 in 3,200 is our cutoff value. So if we look the first row, everything is we see the antigen is settled in the bottom. So this is for the negative absence of any antigen antibody reactions in the sample. So the second row, third row, fourth row, it's the same as with the controls. So it is a positive samples. So this is actually it's read by neck dyes, so it easily interpreted. When we come to our results, the diagnostic performance, so our source of samples will be was well cases and CL cases, but when we see our in-house DAT antigen and the freeze-dried DAT is commercially purchased and RK39, so our DAT is highly sensitive and detects almost more than 97%. In this, when we compare with the two test methods, our test is performs well. But when we see the RK39, it has some cross-reactivity with other, including CL cases and samples from different sources. This is actually, it may be, it's just that the possibility of sharing some genetic similarities with this. So when we compare our, when we see, this is just to see, to show the sensitivity and the specificity of our DAT, in-house DAT and freeze-dried DAT with RK39. So our DAT is has a sensitivity of more than 97% and specificity of more than 98%. So all these actually our DAT is comparable with both freeze-dried DAT and RK39, but RK39 is less to detect the veil. So when we compare our DAT with other cities from East Sudan and North Sudan, so our in-house DAT is higher from this data study performed in these countries. And actually the difference could be there is a variety of sensitivity. So the one of the maybe the possible reason could be the strain variation that affects the level of expressions, and also the maybe the difference in manufacturers. And our data is also comparable with studies done in Sudan. And as a general, our data and freeze-dried data work well and able to identify the subjects correctly. This is to show the agreement between our in-house data and freeze-dried data. So our data is well agreed with the commercially available data with kappa of 0.962. In conclusion, the use of in-house data is demonstrated high performance compared to the freeze-dried data and RK39. So this in-house data can be considered as a valuable tool for the zero diagnosis of VL in endemic areas. So this could make access to VL diagnosis more equitable and enable to be self-sustainable our programs. This all. Thank you.
Okay, thank you for your nice presentation. It says validations of the DAT test. How did you validate your technique? Did you determine the intraday and the interday precision when we develop your method? I didn't see that part.
Actually, when I said validation, just based on the performance of the tests. So our tests, our in-house data performance well as compared to already in with just methods or tests in commercially available. That actually this commercially available techniques or tests already validated. So we compare our in-house data with the already validated techniques.
Thank you so much. Again. Maximum 20 minutes. Coffee. Maximum. Right. So. Okay, now we call on all the speakers. We have arranged a seat for you. We want to listen to you. You are the guys now, and the future scientists. Come on, and you take questions from the audience and share your visions and so on. So much. There's one. Marcos. Marcos. Okay, we take it easy. So we take questions from the audience here and maybe also from our colleagues online, and any clarifications needed and so on. So the audience, you can ask questions, but address it to the right speaker. So you take notes in case you have many questions and so on. Where is Marcos now?
Okay, thank you, Professor, for giving me the opportunity. My question goes to the last speaker, the in-house DATS. You used TB specimen as a control while you do the diagnostics reagent for parasites. What is the theoretical basis of using bacterial specimen as a control in parasitic disease like Leishmaniasis? That's one of my curious questions. My second question is, have you done Ragnus test, which is a test conducted in different laboratory by different laboratory technologists or tester, intra and intraday, and all these things? Because you see, the reliability test is very, very important to ensure as a substitute for commercialized DATs. Thanks so much.
Right. So then who else is all right?
Okay, thank you. My first question goes to Marcos. You have different compounds, about 400 compounds from the pathogen box. Are these compounds soluble, all of them soluble in a particular solvent? What solvent did you use for the test? And why did you choose 1 micromolar for primary screening? What is the base to choose that particular concentration? And the other question is for Dr. Abdurrahman. I think, in your case, you synthesize different analogs of the drug to improve the pharmacodynamic aspect. Did you also consider the pharmacokinetic aspect when you modify when you modify the functional groups? What about changes in the pharmacokinetic aspects of the drug? That is also very important for the efficacy of the drug. Thank you.
Okay, thank you. My question is for Dr. Abdurrahman. So when you were synthesizing different analogs of fexinadazole, at some places you were bringing some big structure or ring from another clinically active drug and mixing with your drug. So if you successfully come up with a new analog which is effective, whose analog would the new product be? Because you are using 2 active drugs and mixing the structures. Wouldn't it be completely a new analog, a new drug? Can we still call it an analog? Is my question. Thank you.
We have about 4, right? But maybe let me add. So if a drug is So they found this toxicity in liver toxicity, wasn't it? Find it in heart. So I suspect it won't be disease specific. So that's one question. The second question is, so fexinidazole was found to be very potent to clear parasites, initial stages, but then this relapse. Okay, so in fact, initially it was thought, based on clinical observations, it was thought fexinidazole is the best because of the parasite clearance. But then the surprising observation is what you simply told us, relapse. How do you explain this? Okay. Any other questions? We will have more questions. Okay, good. Who wants to start? So there are 2 mics.
Okay, so I'll start. I don't know if I miss other questions, but 2 questions were raised. One is about the solubility of the compounds. So these compounds, all 400 compounds, including the reference, were provided in a frozen DMSO solution. So 10 micromolar, 10 milliliter in 10 micromolar solution, millimolar solution. So we made the next dilution with 190 microliter of DMSO, and then the subsequent dilution were made using the culturing media. So all were soluble. The other is why 1 micromolar. So it's just, like screening. The whole purpose of this screening is to get into the clinical trial as quickly as possible. So you don't have to waste like many time by like having a higher S50 and like in testing this compound. So in order to get into the clinical trial, you should have a compound with a minimum S50 value. That's why it's 1 micromolar, and it's also recommended by the donors, the MMV. So they want us to screen all the compounds at 1 micromolar only.
Okay, thank you. Just the issue raised why you include TB as a control to assess this diagnostic performance. Actually, just we used surface antigens, whole surface parasite antigens we used for preparation of our data. So this surface antigen may be a capacity or ability to pick any antibody produced against any infectious diseases. So just to see this sensitivity, we include TB because of we used surface whole antigens. That's why we include TB as one of infectious agents in. For our so sorry for that. Actually we did after we prepared our data antigen, just we sent this data antigen for checking diagnostic performance in Sudan, in Afat University, because they are actually known long lab established lab in this production of the data setup. So we sent for checking our data performance in Sudan. Otherwise, we no locally we didn't, but we checked repeatedly in different samples in our lab for reliability. Yeah. Actually, sorry, Professor, just our cutoff value is 3,200. So we used this cutoff value to say. Yeah, yeah. If actually that should be invalid result, so it should be repeated because it's less than our cutoff value, and it is in between negative result and yeah, but our cutoff value for to say just negative is 300, 3,200. So if just below this and greater than 400, it should be we don't take as negative or positive. So it should be repeated again after some time point.
Thank you. So I think 4 questions has been read. I tried to answer them. So first, it was Dr. Alamu's question regarding the pharmacokinetic modification that we tried. So from this pharmacokinetic parameter, the most important one that was very concerning peculiar to us was the first one with the metabolism, because fexinindazole has a sulfide group, and sulfide-based drugs are very well known to undergo very rapid metabolism. And it is a case with fexinindazole because after the administration, within 1 hour, you cannot find fexinindazole in the patient or in the human. So we wanted to improve that one. So in the analogs, for example, I tried to replace the sulfur with carbon analog because carbon analog is also more metabolically stable than the sulfide. So we tried to address the metabolic stability with fexinindazole. So from this admit, we tried the metabolism. We also were concerned with the with the hydrophilicity also, because whenever I try to modify, the hydrophobicity was, for some analogs, was increased, and I was parallelly synthesizing analog that do have lower water solubility because I wanted to test them, or I didn't want solubility to be a problem for further investigation. So yeah, we have addressed the peculiar metabolic problem and hydrophobicity that may affect the absorption of the drug also. So we did try that one. Regarding Shabru's question, so all of the analog we synthesized. So here we have reported 22 analogs, and all of them, with the exception of one, all of them are novel compounds. If I got your question, it was about novelty, yeah? Hmm? They are novel, they are new compounds. With the exception of one, all of them are new. So the reason I was mentioning is because, for example, I was mentioning about nifortimox, acoziborol, and others. The reason is, so when, as was mentioned earlier, so drug discovery, if you have the target, so you can have the crystal structure of your target, you can try to do the docking so that you can do such kind of drug design strategies. But in this peculiar case, we really don't know the real biological target of fexinindazole. So we need, the other option we have is, in medicinal chemistry, there is something called systematic drug design, or you can call it rational drug design approach. So for every modification you do, you need a reason behind. Targeted some of the of the similarity, biochemical machinery similarity in between trypanosoma and Leishmaniasis. Try to do some hybrid, try to check. So I'm emphasizing novel analog, but it would have some structural feature from fexinindazole and some from already published drug. For example, we also tried to put cyanide because of the toxicity of the nitro group. So all of them are novel, 21 of them, but yeah, that was the. So when you come to Professor Asrat's question, so the first one was hepatotoxicity, if it was detected in mice. So I didn't read that one, so I'm not really sure. But for example, when this drug was first tried, it was for human African trypanosomiasis. And this was for 10 days. So this hepatotoxicity was reported when they were trying to repurpose fexinindazole for Chagas disease. So for when they do that, they use the dosing schedule for benzimidazole, which was 8 weeks. So it changed from 10 days to 8 weeks. And when this happened, this hepatotoxicity, like the increase in ALT and AT was observed. So maybe that could be the reason. So that was what I can say about the liver toxicity in the mice. Maybe they missed it in that regard. So the other one is regarding the relapse associated with fexinindazole. So there was a recently published paper, I think 2022 or 2021. So they tried to answer the reason behind why there is relapse when we administer fexinindazole. So to summarize that, they said the parasite level that remains at the fifth of the 56th day of the clinical trial is very important, that it can tell us whether the drug can produce relapse, whether there will be relapse associated with that treatment or not. So they try amphotericin, fexinindazole, miltefosine. With amphotericin and with miltefosine, the parasite was completely cleared at the 56th day. But in the case of fexinindazole, some parasites were remaining at the 56th day. So they think this parasite dynamics has got something to do why there is relapse, because fexinindazole cannot clear parasites after 56 days of the clinic. I mean, why that is, it would be, for example, if I in the fexinindazole cannot kill intracellular amastigotes, can kill the axenic amastigotes, but when we tried it, even in our case, it was not able to kill the intracellular amastigotes. So maybe during some treatments, there may be some amastigotes left, and that could be the reason for the relapse. Yeah, yeah.
So, you know, it's failed. This one comes spice up. Is it a metabolite that's active against the
In the cell phone. So far, it's only 2 metabolites are reported, and they involve the oxidation of the sulfide group, and only the sulfoxide and the sulfone are reported so far. So this paper was published in PLOS. I think it's 21 or 22. So they think this parasite dynamics. So they did their own trial. Yeah, that's what I was able to get. I think it should be shared. Yeah. Okay. Yeah. Yeah, that's so the only molecular mechanism is reported is so far I can get is so they substrate for nitroreductase 1. So they, because of that, they think they have this reduction pathway, the mechanism responsible. So that.
So one question maybe just for, not that this is not a challenge, because when I see the slides, the plates, so the first 4, was it 3, all agglutination, the rest dots. It would be nice to see the titers, the endpoint titers. Like for TB patients, what is the endpoint titer? For Leash, did you get the endpoint titers or not? Because this is semi-quantitative, it's not a qualitative one. You converted it as a qualitative test by using the cutoff, like positive, negative. Otherwise, you can give a titer. So the slides, of course, you will have many slides, plates. But if I look at that slide, positive sample, with all agglutination max, and negative samples always dots, with not seeing an endpoint. Sometimes you have to suspect what they call prozone, post-zone phenomena. So you get everything, for instance, agglutinating and so on. The point is, it would be nice to see the endpoint titers. And just for the sake of the audience. Okay, this is not a question, just a comment. Okay, and we take an. Okay. Okay, you are in the audience. Sure.
So from the virtual audience, there are 2 questions. From Hassan, this is, I think, for everybody here. There are many drugs for leishmaniasis, but they are too expensive. How do we develop drugs that are effective and also cost effective? Yeah. And to the scientist, and this is from Moses, to the scientist from Uganda, what will be the strategy in creating awareness about the existence of visceral leishmaniasis in the country? Personally, he says that he believes these infections are there, but are often mistaken for other infections and sometimes witchcraft. So how do you guys think you should create awareness? And I have a couple of questions for Abdul. And this is personally my question. So I saw that for one of the problems with the, I don't remember the name, is it fexinindazole? Okay, so is that it's toxic to the liver. And in your assay, you used the HepG2 cell lines, I think. I don't know what the other one is, but HepG2 is a cancer, colon cancer cell line. So I don't, did you think about maybe using primary hepatocytes? Because cancer cell lines are resistant to many drugs that they don't die easily. So that's one of my questions. And when you do this assay, did you also use fexinindazole as a positive control to see if it's actually toxic to those cells and not so much toxic? And your analog is not toxic for those cells. So did you use it as a control in that assay? And if you can explain what ERG means, I know there's a transcription factor called ERG. I know you measured ERG activity, if you can elaborate what that is. And another one, yeah, I think that's pretty much my question. So thank you.
patients. So maybe was even that was treated pediatric. So now treated within, except those who are close to the
The Pokoti group it has about 100,000 people around. So that is the catchment area. And with a reported 0.1% prevalence in the area, could be probably around 10 people would be going over to the nearby. Awareness. Yeah. Some people call it witchcraft, and it is true. Those are some of the challenges that control is facing, and many of them die, and it is witchcraft reported. The good thing is witchcraft is not included in constitution as the recognized, but that is what locally is reported. Now, sensitization one, a person should be familiar with the place, is trying to encourage the research institutions to push further to the region, because even those who are there are part-timers. So if someone just goes there for a week, then leaves, so you cannot have sustainability of support of the region, but rather increasing capacity to manage the infection by allowing health workers live in the place. Then regarding cost effectiveness, still this is a challenge because whenever medicines get out of stock, this particular group of people cannot buy drugs on their own. So coupled with number one, the pains associated, then you have to buy. So you have to convince a person to adhere, but at the same time, there is cost implication. All those are challenges. But if I have this interaction with the colleagues, then I see what is happening here and then in Uganda. I believe if local scientists are empowered, then that will be the solution to developing newer drugs that typically are cost effective, because there is good progress in here. And when you look at the studies elsewhere, we are basically doing prevalence studies, trying to find out who got sick, then what, which medicine can we give? If it is not working, then we have given up. But if there is a program, someone has gotten resistance, we get the lab, we have to optimize treatment, that works very best. But in other areas of research, they are doing well. And we have seen management of infections, particularly bacterial infections, being supported. But for Leishmaniasis, that's what I believe can be done. Thank you. You have to engage local. No, you engage local leaders, because the moment the myth in the country is when someone has been bewitched, you do not inject. That is the first belief. So to pick the person from the community and the treatment is injection, you have to first convince the community. Once the community allows, that is the starting point to management. Yes.
difference. And you have to take all the actions. You know, challenging the community's beliefs aside. So that's what I did. There are many questions. One question is about
Yes, this, yeah, feasibility. Yeah, so I would try.
right. So that's for everybody.
Yeah.
Yeah.
Not just for you.
I would only try to answer from in relation to fexinidazole. For example, fexinidazole, if you see the structure, it's very simple structure, so which makes the synthesis very easy. So, for example, it was 2-stage. Now we can do it from like very cheap starting material. So whenever the synthesis of that drug is very easy, there is a chance that the cost of that drug would be very low. So if you see the others like amphotericin, they're too complex. So that could be one of the problem why they're. So we should, for maybe in this angle, we can focus on less complicated scaffold, which makes the synthesis less difficult, which makes the price of the drug kind of low, so which can make it feasible. So the other one is peculiar to your questions. Yeah, so the first one is, do you use fexinidazole as a control? Yeah, in fact, we did not only for the hepatotoxicity assay, we use it as a control or during microsomal incubation, for the S9 cell incubation, for the HERG cell incubation. So whenever we do, we use fexinidazole as a control because we also wanted to know the stability, the toxicity, and everything in relation to fexinidazole. So we use fexinidazole as a control. And of course, we use miltefosine as a control for the activity also. The other was regarding the primary human hepatocytes you mentioned. So we select TAMA cell line and HepG2 cell line because there are numerous reports that have been very effectively used in order to evaluate the hepatotoxicity profile of various drugs. So we did it also during my PhD because it was also hepatotoxic drug. So they have been well used, so that's why we use them. We're also planning to use primary human hepatocyte cell lines, but they're kind of difficult to handle compared to because they will die easily. So you need expertise, and I'm a synthetic chemist. My focus is only synthesis. I have to learn MTT assay because I have to evaluate my compound. So they are difficult, and it would have been ugly. So, and they're also a bit expensive than the HepG2. So that's where the reason. But we are thinking doing those one also. The other mention is what's HERG. Yeah, so HERG it's human heteroego-related gene. So it's expressed for the protein, for the potassium channel protein, Kv11.1. So those drugs are very important for the action potential of the heart. So if the activity of this drug is inhibited, then from the QPRA signal, there will be elongation of this signal. And this would affect the rhythm of the heart. Even there would be, for example, there are some, I forget the name, a dysfunction in this enzyme can lead to very severe heart conditions also. So now it's like carcinogenicity. Whenever you develop a drug, it's like a very important thing to check whether that drug affects HERG channel or not. So if it's inhibiting this HERG activity, then it cannot pass to the next stage. So it's very important to evaluate HERG activity. So that's why I was surprised, because when I see their material submitted to FDA, so they reported only the cell phone inhibits this HERG, and its IC50 value was low. But maybe our assay is a bit different from them. We use Philippe potassium assay kits. Maybe they use a different technique, but in our case, it does inhibit HERG channel, and this is really problematic because it may lead to, if the patient is taking, then it may lead to some heart problems.
Can I add one?
use as a sensitive one. Oh, was okay. Oh, okay, thank you.
So can I add one point? Here. So it's about the question. Audiences? Or how can we come up with effective and less costly drugs? So I think, you know, we caught some famous quote in Africa. So we said, when there is a political problem, we said African problems with African solutions. So I think it should goes to this kind of problems, like drug problems, science problems. So I think the African nations should invest on drug development capacity in research and development. That's one way to have a good, efficacious drug and also cost-effective drug. The other is we scientists should look for an expedited drug discovery process. So whenever we minimize the time taken to develop a drug, we also minimize the cost it entails when it's produced. So I think we should focus on that too.
Thank you. So reduced R&D costs. Okay. Anyone has a contribution on this, on cost and
how do you see the production of a drug? Let's say you have a drug, and how do you see that? Can this be done here?
Yeah, that's the problem. We cannot do it.
That's a problem. Yeah. In Africa, not more than 20 facilities that are capable of making medicines. And most of the products are not even qualified or pre-qualified by WHO. So I mean, it's a general problem here.
I mean, we shouldn't forget that malnutrition and HIV are drivers of leishmaniasis, both depressing the immune system and exacerbating the disease. So probably most people in this room, if they got bitten by a sandfly, would not develop disease. So, you know, there's a societal situation as well that needs to be addressed.
Any questions for that? Okay, now the last set, whatever number, a couple of questions, and we'll be done then. Anyone has a question? No? Okay. Why not?
Okay, thank you for your nice presentation. I think for my question is for Marcos. I think you have screened 4,400 compounds, and from them, I think you find around 7 for based on the promastigo. So, do you think some more are missing if you start from the axenic assay? And I think most of the time the axenic ones are preferable for screening. And the second one is, I think everyone could reflect on this, including Professor Asrat. I think there are 2 types of species in our country, from the northern ecotype and the southern ecotype. So, Marcos, what type of from which ecotype do you prefer? And do you think that the efficacy, everything could be different if we use the other types of ecotype? I don't know exactly from which ecotype you choose the strengths. And yeah. And do you think that if you use the southern, what will be the problem, or what we use if the northern ecotype? I think there is a real difference that I understood today. And for Dr. Abdurrahman, fexinadazole. I think if it has passed and if it's reached for the clinical trials, and normally just the liver toxicity and how could it reach to that level, having all these problems? Or is it really a problem? Sometimes we may report different adverse effects with different magnitude. So what was the magnitude level, or does it really bother some problem so that it can limit its clinical efficacy? Thank you.
Just help you. Did they had a phase 1 study on fexinidazole? Yeah, or not? No, but, so because phase 1 was for heart situation, where was the phase 1 healthy individuals? Right. But the phase 1 in healthy individuals, did you see any data about it? Or so this is helpful, in terms of hepatotoxicity. So the first observation was in mice, right? The toxicity.
So it was, for example, it's approved by AMD. So fexinidazole was first approved by EMA, or European Medicine Agency. So during its approval, for example, on their websites or on the leaflet for this drug, it says it shouldn't be used in patients with symptoms of liver injury or heart-related problem. The other thing we didn't mention is it also has something to do with neutrophil. It would also affect our neutrophils also. So, so this problem is known, but since for human African trypanosomiasis, it's given only for 10 days. So when it's approved for heart, this was not really the problem, because it's given only for 10 days and not many problems were observed during that one. The major problem was observed when this drug was used for longer time. Like, for example, when they were trying to repurpose this drug for Chagas, they have to use it for 8 weeks. So from 10 days to 8 weeks, you have to give the drug for longer term. And at this time, so when the ALT was seen, so it was too much. So they terminated the clinical trial of fexinidazole for Chagas disease. And now they're trying, so in their paper published, they say this is the reason. Their next target is to try to repurpose this drug, but using the same drug schedule used for heart, not for Chagas. So this could be the problem. The other one is this drug is it's a nitroaromatic drug. So nitroaromatic drug, in modern drug discovery, they are one of the discarded scaffold. Because of that reason I mentioned, their metabolism leads to very reactive nitrogen and oxygen species, which are toxic to the human also. But likely this drug, yeah, only 3 drugs to my knowledge are approved in modern drug discovery, which to have this aromatic nitro group, which are delamanid, proteomanid, and fexinidazole. But they have their own problem also. For example, for delamanide, now it has reproductive toxicity also. So similar drugs, nitro drugs, DNDi was doing study. For example, they are now being terminated because of similar toxicities. So it's not surprising that, there is some toxicity problem with this drug, but yeah, we have to find alternatives so that we can reduce this toxicity. So that's why I was proposing to do this project. There was another problem.
Okay. So I said this before, the promasticot and amasticot, they do have a structural difference. As you can see it from one is flagellated and then another one is not flagellated. There is inherent difference, and there is a difference in the genetics too. So we can say that one drug active in promasticot might not be active in promasticot because there is a bunch of difference between them. The other is their metabolization and the growth rate. So the promasticots are like multiplying rapidly. So they are more prone to growth limiting agents, and the mastigot might not be as prone as pro-mastigot. So these are the differences that we should take account when dealing with the pro-mastigot and amastigots. But different literature says that the probability of getting a good drug candidate, a lead compound, is better when you screen the drug in amastigot stage than the pro-mastigot stage, because the amastigot stage, whether it's axenic or intracellular amastigot, it is similar in the intracellular amastigot stage found in the humans. So you might have a good probability of getting the right candidate. The other is what kind of strain. So there is a difference in efficacy of available drugs in different parts of the region. We don't know exactly what the cause is. It might be the host genetics, it might be even the parasite genetics. I can say quite sure about it, but we use a clinical isolate, which is diagnosed at the LDL, Leishmaniasis Research and Diagnosis Center in Addis Ababa, and the patient was coming from Ambo, I think. That's what I can say. Incutaneous, yeah. Incutaneous and the paper I present is about Etopica. Yeah.
You are right. So for everybody, you really focus on Donovan, you want to test it, use two, north and south. And you have access also to testing in front of you.
Is there a possibility of a mixture of these two species of parasites, north and south, to occur in patients? Or maybe hybrid?
I can answer this. It's a geographic isolation. So usually not, but you know, in the event somebody travels, or vice versa, you know, but these are really anecdotal, but generally it's a pattern, isn't it? So people move and it's a local transmission. So the rare cases could be the case, but last time we discussed the possibility of developing a test to distinguish between the north and the south.
Somehow they have developed some immunity. Can we have some patient may not have clinical symptoms? I don't know.
You live there, you develop immunity. You don't, if you don't live there, like people from the highlands travel there every season, they are very susceptible.
Vaccine development? Why? How come?
There's a lot of natural vaccination, but all the experimental vaccines so far, they have. There's a whole lot of experimental vaccines, but so far nothing has come out. Yeah, there is a serious gap concerning gaps, but naturally, if you will be around the northern region, and if the guys who were repatriated from Sudan and so on, and they were settled in that region, they live in the endemic area, and the incidence is very low. But those guys traveling from Gondar, Tigray, Gojam, and they go to those places, they get VL. They're not exposed so much, and they either live there or encounter the sandfly multiple times and slowly we develop immunity, natural immunity. Mostly, yes. Always exceptions, okay? Not making generalizations, but the relapses, even patients who are treated successfully, they don't get infected again. So there's drug-induced immunity. So they're always at 5%, individuals. You can expect this in nature. All right, we should be coming to an end. So this has been an interesting session, everybody here, and thank you, our colleagues from Uganda. Uganda has been part of the DNDi platform. They were carrying out trials on SSG, PM, but also, was it Ambisom as well? Yes, so Uganda is a good partner for the region here. So thank you, the panelists. Thank you so much. This has been an interesting session. Please give a round of applause to our scientists. So this brings the session to an end. If there's any housekeeping. So tomorrow's session is a bit tricky. I don't know how it goes. So there is a visit to the lab. Tomorrow what happens is you are actually spiking your cultures with LMR blue, right? And waiting 4 hours. Hoping you have done it very well. It was a challenge, a lot of pipetting and so on was, we can expect a lot of errors, which is okay. Right, so that's what happens. So in the morning, maybe spike it well, and you wait a couple of hours, and then you read the results. And then afterwards, it's coming here, there's lunch here, and then the closing sessions. What else happens? Certificates will be offered. Oh, yes, important. Do we get certificates? No? Okay. Why not? I learned a lot. I'm still a student here. And particularly so it's about drug discovery and so on, which is quite new for many of us and myself particularly. Okay. All right. So you can advertise any announcements you have about transport and so on. But at this junction, I stop here. I close the session. Thank you so much.
For now or for tomorrow? Okay. For tomorrow, for those who have a practical sessions, so there will be a car in the Grand campus. Yeah, so you can come here for lunch. I will arrange a car.
Get a transport here. At what time?
At what time they will finish for the practical? Yeah, in the morning, for those who don't have practical, so they can come here and stay. You can work your own. Yeah. So yeah, so the room will be open. Yeah. That's it.
Transport would take them to Black Diamond School.
In the morning. Directly, yeah, directly. Yeah. Yeah, you can.
Okay. Good night. Thank you so much.